techniques of detection or new techniques of working will not be enough to achieve what we want to achieve. We need also the organizational growth through organizational innovation, where an enabling environment is created to enable a scientist or researcher or a student to work as he or she wishes. There's a tropical agriculture platform of FAO, which was launched by G20 countries about 9 years back. It is a multi-layer facilitation mechanism to improve efficiency and effectiveness of capacity development. Mind it, capacity development. This is a key word here. And also knowledge sharing to strengthen agriculture innovation system in the subtopic. And there are 47 partners at a global level. And I'm happy to say I'm chairing this tab for the coming 2 years. And what we are doing is we are trying to promote the agriculture innovation system vis-a-vis all the disciplines, which include plant health management. Agriculture innovation, as you know, all know, is the process whereby individuals or organizations bring new or existing products, process, or ways of organization into use for the first time in a specific context, which means you are working on a research output. You have a research output, and how you bring innovation to tailor the output to meet the needs of the farmers or the policymakers or the input suppliers. So here, innovation refers to technologies and practices, which may be technology for improved variety, which may be technology for reduced post-harvest losses, technology for spraying methods, technology for diagnostics, but it also refers to process and organizational reforms or forms like public-private partnership. How are you bringing? How you are organizing farmers and cooperatives to make your things happen? What are the performance contracts? What are the policies available? So we are not to confuse innovation with innovation with invention, and we have to make sure that innovation implies application and change. You have a research output. How do you apply it to bring a change by making a change in your output? This is something what we are now propagating a lot, and this requires a lot of partnership and networking, reflection and joint learning, co-creation of knowledge, facilitation and brokerage, even at the knowledge level, leadership facilitation, documentation of change process. How do you document all the changes that you are bringing in? It's quite an interesting and comprehensive exercise, which make your subject more relevant to the civil society. And if you look at this innovation system, and if you look at more specific with the plant health management, agriculture increasingly involve complex interaction among stakeholders. We know at multiple levels. So we need a system perspective. So is the case with plant health management. What happens? Most of you are here in research and education. You would like to see what you are doing in agriculture research or what you are doing in terms of education. But that is good for you to have your qualification, degree, and maybe a job, but to have a holistic system, how you can move ahead. You need to have a solid interaction with agriculture value chain actors and organization in a business mode. You need to be building the institution, the stakeholder platform. You need to bring agriculture extension from both public and private and civil society. You need to have contractual arrangements. So for all this, what you need is an enabling environment. Enabling environment leads to a kind of policies and investment and informal institutions and practices and behavior. You need a political will. You need science and technology policy under which you work. You need science actors, which you are. All of you are. You all are placed here. And you also need technology from other sectors, which may be from the field of physiology, which may be from the field of agriculture engineering, which may be from the field of food processing or horticulture, and so on. So Plant health management is actually also we need to now look as agriculture innovation system in a very uh, uh, what do you call a system perspective to really make the change happen. And this tropical agriculture platform has brought some common framework, which in a nutshell provide concept, principle, approaches, and tools to better understand the agriculture innovation system architecture. And it helps in assessment of capacity development needs. And practically it focuses on functional capacity. What is functional capacity? It is a soft skill. You may be a good gold medalist. You may get 4 out of 4 or 5 out of 5 or 10 out of 10 OGPA. You are highly respected. You are the best. When you come to your real life, when you come for a job, when you really want to use your knowledge, majority of such people normally have problems with their functional capacity. What is a functional capacity? I'll just tell you. Functional capacity is your capacity to navigate complexity. You come in a world where it's complex. You have your professors, you have your private companies, you have the policymakers, you have farmers. How you really navigate in that complexity, keeping in view your knowledge and background? Do you have the capacity to collaborate? How you make it? Capacity of reflection and learning on what you are doing, capacity to engage in a strategic and political process where you influence a policy issue. So all these points actually go to capacity to adapt and respond to realize the potential of innovation. So this is the functional capacity I was talking before, where we work on with the participatory capacity development. There has to be a capacity, not that you have a summer school, you have a winter school, you go for training, you come back. It doesn't work much. You may go to USA, you may do postdocs, you come back. It doesn't work much unless, along with your individual capacity, we look for the organizational capacity. Is your university, is your institution has the right ambience to support you, to provide you all the enabling environment? Do you have the right policies under which the institution will have motivation to support you? So when we talk of indie innovation, functional capacity is very crucial, where we have to look for both capacity development of the individual, of the organization, and of the enabling environment, the policymakers. And this can happen by active participation and involvement, right from the very junior to the top level management in the university. It may be deans and director of research and vice chancellors. And at the bottom of the range, you may have students and the postdocs and all, where you have to sit on one platform and really make sure, break the barriers of bureaucracy and culture at times to bring the work culture in such a way that you have a holistic capacity development in your organization. Otherwise, nothing will happen. You will make your career, you will go individually, you will have your gold medals, you will get impact factor publication. End of the life, you are not making change in the whole system. There's a lot of role of the private sector. I have listed a couple of areas where private sector needs to come forward. I'm not going to read all, but these are the areas related to the plant health management, but private sector can play a very crucial role. I may show my PowerPoints. With all respecting the time, I may like to come to the end of my meeting. I may not speak much. There's a role of FAO, a different platform. In Asia, we have the Asia and Pacific Plant Protection Commission. So they all are playing their respective roles as much as they can. What is important is working locally and at national level, and then to take help from the regional organization like this, and then to work globally. There are a lot of international efforts which have been done. I have listed a few organizations which have come up in various ways to help, and we are taking help of some of these organizations in our working to see that we make the right technology or right innovation approaches reach to all the partners in South and Southeast Asia. So there are regional bodies, like I have listed a few here, which includes APARI, where I'm working. We are doing whatever we can in terms of capacity building, research and development, and as a hub for knowledge management. And if you look specifically to APARI, we had a lot of programs in the last 3 years on capacity building, R&D, knowledge management, and even coordination and networking. And mainly R&D in plant protection, innovation, and policy. We have 4 big projects working across the region in many countries. And this is what bringing us more satisfaction of reaching to the people with the innovation. And more so, thanks to the best wishes and hard work done by all the partners, we recently got another project from WTO where we are strengthening the phytosanitary practices and the public-private partnership.
ಹಾಗಾಗಿ ಪೋಲಿಯೋ ಡೋಸ್ ಪ್ರತಿ ಬಾರಿಯೂ ಕುಡಿಸಬೇಕು 2 ಹನಿ ಪ್ರತಿ ಬಾರಿ ಪೋಲಿಯೋ ನಿರಂತರ ಹುಟ್ಟಿನ
enhance the seed trade with different partners like Asia Pacific Plant Protection Commission, Asia Pacific Seed Association, CropLife Asia, International Seed Federation in Geneva, and American Seed Trade Association. We are going to work in the listed 7 countries in South and Southeast Asia where we see what global seed supply chain has been affected post-COVID, how we can bring it more into shape. So I would like to stop here because I think my time is over and I would like to respect the time. The journey in the plant health management is a fascinating one. It's a very fascinating one. And the message I want to give, and especially to the younger generation who are the future of plant health management, this I'm telling after 42 years of my experience of working in diverse multi-stakeholder environment in research, development, and policies. And I mean it what I say, there are 2 points. Number 1, be focused. If you are doing research, be focused on research. If you are doing extension, be focused. If you are doing in development work, if you are in teaching, be focused. But please have a 360-degree view of the subject. Be focused, go vertical to excel. But you should be knowing the relevance, the context, what other people are doing, and how you can find partnership, how you can make your work more interesting, more synergistic with all other players around you. Secondly, which I try to follow in my life, I would like to say very strongly to every younger generation is, please discover yourself. We human beings don't know our own potential. We don't know what capacity we have. Every human being has a lot of gene silencing. You have to wake it up. Discover yourself. Accept the challenges. You may be transferred to a remote area. You may blame your professor or the vice chancellor or director. No. Take every challenge and make the best of it. You may be put from research to education. You may be put from education to extension or extension to some office work or some technical work to support the senior management. Every opportunity you should accept the challenge. If you accept the challenge, I think your brain opens up more if you accept in a positive way. And you, end of the day, discover yourself. It is you who is important. If you feel that you can do, I think the whole field of plant health management is bound to benefit a lot. And with these words, I would like to stop here. My email ID is here. If anyone wants to contact for anything about job opportunities, future, or global working, I'll be happy to see whatever I can do. But I would like to acknowledge before I close to Indian Society of Mycology and Plant Pathology, which gave me a chance to be the president and share my thoughts with the members of ISMPP. And all my thoughts, I think, cannot come alone. I'm a very small molecule. I'm very indebted to all the organizations which gave me a lot when I worked with them, right from my academic career to from JP Pant University, Pantnagar to NBPGR to NICR to INGRA, France, where I did a PhD and postdoc, and to KEBI South Asia, and now to Apari Bangkok. And also a couple of consultancies which I have done was a big learning experience with some global agencies and other societies where I had been president, like IPS or INSOP from Ludhiana, and where I had been vice president, like Plant Protection Association or Indian Society of Plant Genetic Resource. And finally, my very special thanks to all my professional colleagues who helped me to reach where I could, and my personal family, my children who sacrificed really, and my wife who had to live more than half the life without me because I was more absorbed in my career. I owe everyone a lot. Thank you so much. And these are the list of the logos of Apari partners with whom we work. Thank you. Thank you, one and all.
Thank you, sir. That was an enlightening and informative presentation. Now let's move on to the first session of the day, that is ISMPP Award Competitions, which comprises PP Singhal Memorial PA Industries Award Competition and Guman Devi Verma Memorial Best Woman Scientist Award Competition. This session is being chaired by Dr. Suresh Ekbote, Dean, College of Horticulture, Hiriyur. Dr. Suresh Ekbote has been extensively involved in teaching, research, and extension activities for the last 24 years. For his outstanding contributions in the field of plant pathology, he has
Dr. Ekbote, Dr. Suresh Ekbote, are you there?
Mesta, I am here. You are hearing me. Mesta,
Sir?
Mesta.
Yes.
Me.
I
am here.
Okay, we can hear now. Yeah. Please put on your video.
Yes. Ide nodi. Mesta, you see that it is Niharika name. I think it is not. Okay. For time being. Okay. I am here.
Okay. Thank you. Welcome. Please.
So now let's move on to the first session of the day, that is ISMPP Award Competitions, which comprises PP Singhal Memorial PA Industries Award Competition and Guman Devi Verma Memorial Best Woman Scientist Award Competition. This session is being chaired by Dr. Suresh Ekbote, Dean, College of Horticulture, Hiriyur. Dr. Suresh Ekbote has been extensively involved in teaching, research, and extension activities for the last 24 years. For his outstanding contributions in the field of plant pathology, he has been awarded National Environmental Science Academy Fellow of the Year 2020. He is also the recipient of the Distinguished Scientist Award from Society for Scientific Development in Agriculture and Technology and has been granted the Certificate of Merit Award from the University of Agricultural and Horticultural Sciences, Shimoga. The session is being co-chaired by Dr. Kantarasha V, Professor of Plant Pathology and Head ICAR ASCRPN Fruits and Extension Leader, Horticulture Extension Education Unit at Bavi. He has been the recipient of the Best Extension Scientist Award by UAS Raichur and has been granted National Award for the Best ICAR ASCRPN Fruits during 2 consecutive years, that is 2018 and 2019. He has put forth many outstanding contributions to the field of plant pathology and has published 44 research papers, 26 abstracts, and many popular articles and extension bulletins. The reportees for the session are Dr. Sangeeta, Assistant Professor of Plant Pathology, College of Horticulture, Bengaluru, and Dr. Sadanat Mushrif, Assistant Professor of Plant Pathology, College of Horticulture, Kolar. Now, I welcome Dr. Suresh Ekbote to carry on the session. Sir, please.
Good morning to everyone. And first of all, I'll thank Organizing Secretary Dr. R.K. Mesta, Professor and Head, University of Horticultural Sciences, Bagalkot, and also my colleague who is with me, Dr. Kantraj from AICRP Fruits at Arbavi, and also rapporteurs. And I welcome all the participants who are participating for the award competition PP Single Memorial Award for the PI Memorial Award. And the time is 15 minutes. 13 minutes. After 13 minutes, you will get one bell. So another 2 minutes will be left over. And I request all the participants who are participating for the award competition to restrict themselves the timing 13 plus 2. And I also request all the judges who are there, you strictly monitor the timing 13 plus 2 minutes. So it will be belled by the central there from the Bagalapur. So you be cautious about the timings. I request one by one the participants as to present 13 minutes plus 2 minutes. Okay. I think rapporteurs take care of the timing and also timers. And also the co-chairman, Dr. Kantraj, can you speak a few words before starting of this one?
Yes, sir. Yes, sir.
Please. Yes, go ahead.
Very good morning to one and all. First of all, I thank Dr. R.K. Mista, organizing secretary, and his team for giving me this very wonderful opportunity to participate in this. So I wish all the best for all the participants for these two prestigious awards. So yeah, I thank Dr. Ekbote, the Dean COH Irio, and I once again mentioning that the participants please follow the guidelines and what are the time restrictions they are given. So I once again wish you all the best for your presentation. Thank you, sir. Thank you, Dr. Ekbote, sir, Chairman, and Dr. R.K. Mista, sir. Thank you.
Thank you, thank you. Chairman, sir, I have one query. Here, 4 participants list is given to you, right, sir? Chairman, sir.
Yes. Yes.
Yes.
You call one by one, no problem.
Here, first, the Praveen Satpute.
Yes, sir. Yes.
You just inquire all 4 are there or not? Just one.
Whether Praveen Satpute is there?
Yes, sir. I'm here.
Dr. Chairman, sir, please note that he is going to first. He is going to present first.
Okay. Praveen, go ahead.
Dr. Mohidwin.
Yes, sir. Yes, sir. I'm here.
Yeah, he is going. He is the second contestant. And Dr. Gurudat Hegde.
Yes, sir. I'm also there. Good morning.
Yes. Good morning.
Dr. Devendra Jain. Dr. Devendra Jain.
Devendra Jain is there.
Okay, it seems he is not there. After the 3 contestants finish, let him present. Chairman, sir, please, one more request. Can you hear this bell?
Yes. No problem.
After 13 minutes, one reminder will be given, sir. And in another 2 minutes, they have to complete.
Complete.
Okay, sir.
If it is exceeded, then negative marking will be there. It is for information.
I told all the judges to note down all the things.
Yes.
Okay,
sir. Chairman, sir, it is over to you now.
Okay, Mr. Praveen, Dr. Praveen, can you go ahead?
Yes, sir. Hello, everyone, sir. Yes, sir. Yeah, sir. Very good morning, everyone. I am Dr. Praveen from Karnataka University, Dharwad. The title of my today's presentation is Biodegradation and Molecular Characterization of the Triazole Fungicide, Tricyclaazole by pesticide-resistant Pseudomonas butida, isolated from agricultural soil. So as we know that pesticides are the chemical substance used to kill, repel, or control the many kinds of diseases and pests. So, however, extensive and inappropriate usage of these chemical formulations or pesticides leads to many long-lasting effects like water contamination, groundwater contamination, air contamination, and habitat contamination near the fields. So, increased usage or inappropriate usage of hazardous pesticides or chemical formulations in agriculture systems, especially in India. Why? Because we are the largest agriculture system. We have the largest agriculture system. It is causing the soil fertility and effect on the beneficial microorganisms present in the soil with the toxic chemicals or the residues present in pesticides. This is the cycle of pesticide, how the residues will enter into the different habitats of the environment. So chemicals, how they enter. So farmers will spray the pesticide to the standing crops. It will reach to the soil. Then after the rain, it will go to the nearby lake and it will affect drinkers and aquatic animals or marine animals. It will affect all the habitats in the environment. So next, this is the tricycloazole structure. Tricycloazole is a fungicide. It is a fungicide. It is a group of triazole fungicide. It is used against rice blast caused by the Magnophorte oryzae. Next, I will move to the materials and method. The tricycloazole, 96% is tricycloazole was procured from the Rallis, Bengaluru. Next, the microbial strain, Pseudomonas putida, which was previously isolated from in our lab. So it is a propiconazole resistant bacteria. Propiconazole also falls under triazole group of fungicide. So what we did is initially we acclimatized this Pseudomonas putida to the tricycloazole to the different concentrations of tricycloazole at pH 7. Next, further, what we did is, after the complete resistance of Pseudomonas putida to tricycloazole, we carried out a degradation of pesticide. What we did, we have taken a sterile mineral salt media, which contains only tricycloazole of 4.0 mg per liter, so which act as a sole carbon source and energy source for the bacteria. Further, we have inoculated Pseudomonas putida of 3 into 10 raised to minus 6 CFU per ml and incubated at 35 degrees Celsius at rotor incubator shaker. Next, this is the analytical study. Further, the incubation of bacteria. Next, we have to study the process of degradation of pesticide and how the precursors will produce during the degradation. What we did is periodically 5 ml of culture filtrates were aseptically withdrawn at 12 hours of time interval. Further, these culture filtrates were measured for bacterial growth at 600 nanometer. Further, to study the degradation studies and precursors produce, what we did is we centrifuged the culture filtrates at 10,000 RPM for 15 minutes at 4 degrees Celsius. Further obtaining the culture filtrate, we extracted with ethyl acetate by using a rotor flash evaporator, and obtained residues were redissolved in the HPLC grade acetonitrile. HPLC grade acetonitrile is used because we are going to do LCMS for the identification of precursors produced during the degradation process. So we confirm the degradation of the pesticide using the UPLC agilent and with a UV detector of C18 column. Next, further, we have identified C cytochrome P415 gene in the Pseudomonas putida. So we have designed a primer. This primer we have designed from the Pseudomonas putida locus AF014839 and with the base pair of 1426194. So previously we have presented this paper on annals of microbiology. Further, what we did is for the domain identification, this is a purely in silico work. After the in vitro work, we have carried out in silico work. How this cytochrome P450 acts on the degradation of toxic substance. Further, the sequence of the cytochrome P450 gene was identified through the sequencing and transferred to the protein sequencing using a Swiss gene server. This protein again used for the domain prediction by the SBSC server. This server are mainly used for the domain prediction in the protein sequence. Further, the domain was identified using the BLAST. BLAST is the local alignment search tool. So this is used for the template identification. Further, the sequence which is having the highest identity with our sequence of cytochrome P450 having high score and low or less E value was aligned or taken for the custom W program and template structure production using the modulars. Next, the structure prediction of the cytochrome P450. The structure of Pseudomonas putida cytochrome P450 was built using the modular software. This modular software is specifically used for the prediction or the build of the 3D structure. The three-dimensional structure of cytochrome P450 generated by the minimized molecular probability density function. Next, after the generation of 3D structure, we have to identify what are the possible active sites present in the cytochrome P450 using the CAST-P server. Further, next, the docking system, how this docking system work with isolated cytochrome P450 with the existed pesticide. So the docking was carried out using the GOLD system. This is the genetic optimization of ligand docking. Next, we'll move to the result. This is the AFM result of the pesticide. The first A is the control one without adding any pesticide. So here we can see a clear picture baseline. There is no effect on the pesticide. There is no effect on the bacteria on pesticide. Next, here we can see B, a stunted growth of bacteria during the initial stage of acclimatization. So during the initial stage, stunted growth we observe. Next, at 2 mg per ml, the rupture of bacteria was observed, and we again, we acclimatized the bacteria to the pesticide. Here comes, this is the first A is showing the control. Next, the bacteria acclimatized to different concentration, 4 mg per ml and 5 mg per liter. 4 mg per liter and 5 mg per liter. Here showing the bacteria is resistant to tricycloazole and it is having the capacity to degrade or to consume the pesticide for its growth and metabolism. Next, this is the degradation study. In 96 hours, this pesticide-resistant Pseudomonas putida degraded up to 4.23 mg per liter of degradation. It is investigated and analyzed by the LC. Okay, the precursors or metabolites of TCZ or tricycloazole were identified by mass spectra and tentative pathway also proposed. This is the tentative pathway and LC showing the result, how the degradation occurs. This A, first, this is the standard LC showing the single peak extracted immediately after the addition of tricycloazole into the blast. This is showing how the next B is showing how the degradation is occurs. Here you can see a different TSCs or different chromatograms were observed during the or formed during the degradation study. This C is showing how this pesticide is converted into smaller products. Here we can see the last end product is carbon dioxide and water. So we have totally identified 4 precursors. This is the fifth one is we have procured from the other article by reference. Next, this is the identification of cytochrome P450 in Pseudomonas putida. Here we can see at the 1.3 kDa, So it was identified and it was extracted at 1.4 kDa from the Pseudomonas foetida. So this is the homology model of cytochrome P450. So cytochrome P450 monooxygenase PYKC from the Streptomyces venezuela having the 43% identity with the sequence of cytochrome P450 from Pseudomonas foetida. So this Pseudomonas foetida cytochrome P450 found 43% similarity with Streptomycin venezuela. Next, further, main active sites identified were aspirin, cysteine, leucine, and tyrosine in cytochrome P450 Pseudomonas foetida. Next, this is the docking study. Here we can see that this is the gold score of 2.01 minus 2.01 of gold score. It is docked at the histine and histine 1sp. So from this, we can say that the selected dock conformation of tricyclazole into the cytochrome P450 binding site. The dock's result revealed that tricyclazole is having the capacity to inhibit the compound, tricyclazole of cytochrome P450 in Pseudomonas foetida. From this result, I conclude that we have isolated the most potent biological weapon, which is having the capacity to degrade a wide variety of pesticide or chemical formulations which are harmful to the environment, soil fertility, and microbes present in the soil. Thank you, everyone. And any questions? Thank you, everyone.
Okay, thank you, Praveen, for your nice
Yes.
presentation. Dr. Mesta,
Yeah. Yes, sir.
can you give, I have
Yes,
sent
sir.
message about Dr. PP single.
Sir, your voice is not clear.
Okay, 1 minute, 1 minute. Dr. Mesta,
Sir, your voice is not clear. Okay, he has finished. You can call the second speaker.
okay,
For the
Dr.
information of judges, he has taken 13 minutes.
Yes. Okay. The next participant, F.A. Mohiddin, Dr. F.A. Mohiddin.
Yes, sir. Yes, sir. I am sharing my slides, sir.
Okay, okay. Let us wait. After sharing is only start presentation, then only count the time. Okay, please. Timers. Wait.
Shall I start, sir?
Oh, okay, you start now.
Okay,
Please go
sir.
ahead.
Okay. Thank you very much, organizing committee, for allowing me to present my research under PP Single Memorial Award. Um, uh, Myself, Dr. Fayaz Mahideen, senior scientist working at MRCFC Khodwini, Mote Research Center for Field Crops Khodwini, which comes under the organization of Coast Kashmir. The title of my talk is Evaluation of some combi products for the management of blast disease of rice under temperate conditions in India. Well, the test site was, as I told, Mote Research Center for Field Crops, which is located at an altitude of 1,601 meters above mean sea level. Its latitude and longitude is 33 into 75 meters, and ecosystem, it's irrigated ecosystem where we have laid the experiment. Our place is very much prone as far as blast is concerned. You just see here the blast initiation symptoms. First here you are seeing here first initiation of blast lesions which occur on the side of the leaves. Then it coalesces. The lesions coalesce together, and they start coalescing together and cover the whole leaf. Blast occurs at two stages. When it either affects on the leaves as well as it affects on the neck blast. Means when it is more severe when it affects on the neck. Here you are seeing the leaf symptoms where blast affects. When it affects, it covers the whole leaf and which affects the photosynthesis of the plant. You just see another photograph of here, which is the aerial view when the blast affects on the neck. When it affects on the neck, what happens? All the crop has been lost. What we have seen here, 100% yield loss occurs when the blast affects on the neck. Keeping in view the effect of the blast in our place here in Kashmir, what we have done, we have evaluated some new combination fungicides for the management of rice blast disease in temperate regions. Why temperate region? Because I already told you that the trial was conducted at an altitude of 1,601 meters above mean sea level. And why new combination fungicides? Because mostly my colleague just say that tricyclozole is used against the blast, but now recently it has been banned because residue has been found in lot in basmati rice outside when we exported it. That's why tricyclozole has been banned. And then we study here new combination fungicides as an alternate to tricyclozole. As far as material methods are concerned, yes, we conducted a trial. Field trial was conducted at MRCFC Khodwani, which comes under Coast Kashmir. The experiment was laid under complete randomized block design with 3 replications. The plots were separated 1 meter apart, and there were 8 blocks in each replication, which were separated by 0.5 meters. 25 days old seedlings were transplanted to the main field, and conventional cultivation practices were followed. The experiment consists of 10 treatments, including 9 fungicides and 1 water spray control. These were the fungicides, combination of fungicides which we used in the field in 2018 and 19. 2-year trial was conducted. Flucilizole plus carbendazim, azoxystrobin plus difenoconazole, azoxystrobin plus tebuconazole, tricyclozole plus mancozeb, Zynab plus hexaconazole, trifloxystrobin plus tebuconazole, mancozeb plus carbendazim, fluoxypyroxide plus epoxyconazole, and tricyclozole. Why tricyclozole alone? Because other fungicides were seen as its alternate, because it has been banned now. You just see this is the field view where we have applied this fungicides. We applied 2 treatments were done, one after 1 month after sowing, and thereafter second treatment was done just after technical initiation. This is the PDI percent disease intensity was calculated with this formula, summation of N into Y into 100 by N into G, where N is the number of infected leaves in a category, V is the category value from 0 to 9, N total number of leaves tested in each application, and G highest category value. Well, statistical analysis was done by using MS Excel, as well as ANOVA was done by using Duncan's multiple range test method. Yes, disease rating scale was carried out which was given by International Rice Research Institute in 2003. The category was 0 to 9 scale. 0 no lesions were found, 1 small brown specks, and 2 small roundish to slightly elongated necrotic gray spots were found. And similarly, 9 scale was found, and more than 75% leaf area was affected. Yes, another percent disease control was carried out by taking control minus TE divided by control into 100, where PDC is percent disease control, C is the disease incidence in control plot, and T is the disease incidence in fungicide treated plots. The data in the yield was recorded at maturity state by marking 3 into 10 meter section within each plot using a wire frame as described by Seabold, and tillers within the market area were cut and harvested for estimating the yield. Yes, these were the results which we have seen. The parameters which we have taken here is the leaf blast incidence 1, neck blast incidence 2, leaf blast incidence intensity and yield. You just see, yes, although tricyclozole was found best as compared to other fungicides, but azoxystrobin plus difenoconazole, azoxystrobin plus tebuconazole were at par with tricyclozole in managing the leaf blast incidence, in managing the neck blast incidence, as well as leaf blast incidence intensity. Also, you just see here tricyclozole was found effective in increasing the yield, 67 quintals per hectare, but azoxystrobin difenoconazole was at par with tricyclozole, and azoxystrobin tebuconazole was at par. These two were at par with tricyclozole as far as yield is concerned, and as far as other parameters, means leaf blast intensity, neck blast intensity, and leaf blast incidence is concerned. Here Zainab and hexaconazole was found least effective in managing neck blast in 2018. I told earlier that 2 years trial was conducted in 2018 and 2019. Similarly, in 2019, we found, yes, tricyclozole was found best, but these were at par. Azoxystrobin difenoconazole, azoxystrobin tebuconazole were found at par with tricyclozole as far as the management of the disease is concerned, such as leaf blast incidence, neck blast incidence, and leaf blast intensity. You just see per person disease control was also measured, and similarly here T9 treatment was found best as far as disease control is concerned, but azoxystrobin plus difenoconazole and azoxystrobin phenoconazole were found at par in both the years for managing the disease. Comparing the weather data of 2018 and 19, you are seeing I shown in 2018 the disease was found more as compared to 2019. The reason behind this is that that it coincides with the rainfall. Total rainfall was more, and the disease occurs more when there is a more rainfall in the growing season. You just see here in 2018 the rainfall was more as compared to 2019. This might be the reason that the disease was more in 2018 as compared to 2019. Finally, the conclusion in the current study, blast disease shows highest severity in 2018 compared to 2019, which might be attributed due to high rainfall, as the disease has been reported to be positively correlated with the rainfall. Yes, tricyclozole was found best to be the most effective fungicide in combating the blast disease under field conditions in both the years. Tricyclozole exhibited greatest efficacy with disease control of up to 89.43%, while as least disease control 58.66% was recorded with mancozeb plus carbendazim. The data represented in table 1 and table 2 reveals that the combination of fungicides containing citrobulin and triazoles, means azoxystrobin plus difenoconazole 0.1% and azoxystrobin plus tebuconazole, were always at par with tricyclozole in their activity against the disease. Hence, these fungicides offer a valid alternative to tricyclozole in rice blast management. Actually, the objective behind the study was to see alternate as far as tricyclozole is concerned, and we were successfully managed the disease by using azoxystrobin plus difenoconazole and azoxystrobin plus difenoconazole as an alternative to tricyclozole. Well, acknowledgements. I am highly thankful to both co-authors of the paper, Dr. N.R. Sophie, Dr. Arif, and Dr. N.A. Bhatt, for contributing in the managed sector. Thank you very much, sir.
Okay, thank you, Dr. F.M. Mohiddin. You have taken us to the Kashmir within 15 minutes from wherever we are. A nice presentation and nice work you have carried out.
Hello.
Thank you, sir. Thank you very much, sir. Thank you very much.
Chairman, sir.
High elevation you have carried out. I think if
I got
I'm not mistaken, 1601 mean sea level. You have
Yes,
working.
sir. Yes, sir. Yes, sir.
Definitely very good work.
Thank you.
You have taken us to within 15 minutes to your Kashmir. Thank you for that.
Thank you,
sir.
Sir.
I request all the judges to note down the timing.
Yes, sir. He has taken 10 minutes exactly, sir.
Yes.
Yes, sir. Out of 15 minutes allotted, he has completed 5 minutes earlier.
Thank you, sir. Thank you very
This
much.
is for information of judges. Please note down the time.
Kindly allow me to share the screen, sir.
Okay, sir. Just a minute, sir.
Dr. Gurudat Hegde or
Yes, sir. I'm here.
hello.
Yes, sir. I'm here. Sir, is it visible?
Yes, visible and audible.
Yes, sir. Thank you. Shall I start, sir?
Chairman, sir.
Yeah, Gurudat, please start.
Yes, sir. Good morning to everybody. Honorable President of ISMPP, Dr. Khetrapal, sir. Respected Chairman, Co-Chairman, sirs, and very respected judges for the session. All the distinguished members of the members and secretary of ISMPP, organizing secretary and his team of UHS Bagalkot, and plant pathology delegates, student, friends. Thank you very much for giving me this opportunity to be a part of this very prestigious PP Singhal Memorial Pesticide Industries Award. On this occasion, I would like to give my delivery on influence of weather parameters on development of fungal foliar pathogens of groundnut and management strategies. I'm working in the University of Agriculture Sciences, Dharwad. So my outline of my presentation is first one is introduction and present status of losses due to this disease of groundnut, extent of losses, epidemiological factors, which is a very theme and core theme of this presentation, wherein I'm going to correlate the spore load and weather factors with the different fungal foliar pathogens, early leaf spot, late leaf spot, and rust, correlation and multiple regression in relation to fungal foliar pathogens, and another core theme of this prestigious award is chemicals that we have used for managing these fungal foliar diseases, that is chemical and integrated approaches to manage the fungal foliar diseases, followed by conclusion and Thanksgiving. As we all know that peanut, it's one of the world's largest oilseed crop, which is called as king of oilseeds, which contain 40 to 60% oil, 20 to 40% protein, 10 to 20% carbohydrate, and so it is referred as high nutritive value crop. Largest producer China and India, followed by United States of America. Apart from being a direct consumption as feed fodder, vegetable oil, and also as a source for protein feed in the animal industry. Multiple uses of peanut make it an excellent cash crop for domestic as well as international trade. Despite its immense prospects and undeniable importance, crop disease acts as one of the major constraints in the production and productivity. The biotic factors severely affecting the yield levels of groundnut are fungal foliar diseases like I already mentioned, late leaf spot, early leaf spot, and rust. And we should also know that this is this is the statistics of groundnut both in India and Karnataka. It is increasing over the years, and we can see the symptoms of early leaf spot caused by Cercospora arachidicola, late leaf spot Phaseropsis personata, rust, Coccinia arachidis, and stem rot, groundnut bud necrosis, groundnut rosette diseases are also other major diseases, but I am focusing on only the fungal foliar diseases, which altogether contributes to the 50 to 70% yield loss in groundnut. Yeah, now coming to the objective, keeping in the theme area of this prestigious award, that is one is epidemiology and another one is chemicals in disease management. The objectives have been set like survey for the severity of fungal foliar diseases in northern parts of Karnataka, and to understand the relationship of weather factors with the disease development, and then management strategies using chemicals and also a biocontrol agent as an integrated approach. And we can see all the pathologists are familiar with all these kind of spots which are there on the upper surface, and on the lower surface we can see the rust leaf spot and early leaf spot in a groundnut crop. Now to understand the severity of the disease which was conducted during the survey during 2018, see we can see among the different districts surveyed in northern parts of Karnataka, it is very clear that the early leaf spot, the late leaf spot, and the rust were very prominent and very severe in Dharwad district, followed by Haveri, then Belgaum and Bagalkot district. This is mainly due to the variety grown in the locality, and it was also observed that the disease severity was more in local and susceptible genotypes like JL24, TMU2, Dhanalakshmi, and other local varieties. Whereas improved varieties GPBD4, G2-52 were found to be resistant to foliar disease, which is reflected in terms of less disease severity. And we can see here the farmer's field, wherein we can see the infection of these fungal foliar diseases. Also, it is reflected in the graphical representation, which signifies the higher disease incidence in Dharwad district. All the three were more in Dharwad district. And coming to the objective two, that is understand the weather factors with respect to the disease. Here we have used the highly susceptible variety JL24 and we have sown it in 3 different dates of sowing at 15 days interval. A spore trap was used for aerobiological studies and weekly PDI spore load weather data was used. And we can see these are the 3 dates of sowing: 22nd June, 1st 7th July, second date of sowing, and 24th July was the third date of sowing. And weekly spore load PDI was calculated. We can see the staggered planting at 3 different dates of sowing. We can see the installation of aeroscope and also the slides, glycerin, Vaseline coated slide for the to know the observation on spore load. And a standard protocol were used to assess the PDI based on the scale, leaf spot and rust was assessed. And see, this is a very important weather parameters wherein we can see the meteorological data and the spore count at various point of its observation. We can see the spore load was at its peak during 38th standard meteorological week for early leaf spot. During the previous week, maximum temperature was 29.1, minimum temperature was 21.4, and rainfall was 40.2 millimeter with 2 rainy days. Morning and evening relative humidity varied from 96 to 81 respectively, and wind speed was 5.03 kilometer hour. We can see here first date of sowing. D1 is first date of sowing, D2 second date of sowing, D3 third date of sowing. With respect to the early leaf spot, we can see the maximum incidence in the first date of sowing, that is 45.1%, and further it declined to 30 and 27.3. When we come to the next disease, late leaf spot, it is also reflected that a similar trend was observed, that is D1, D2, D3. The maximum incidence of late leaf spot was observed at 55.3, that is first date of sowing. Further, it declined to 48.5 and 42.5. And the spore load of rust was at its peak during 40th standard meteorological, which is highlighted there. During the previous week, the maximum temperature was 28.4, minimum temperature 20.2, and rainfall was 82.2, with 6 rainy days, morning and evening relative humidity. It varied from 88 to 96 respectively, and wind speed was 4.6 kilometer per hour. This has significantly influenced the spore load and outbreak of the disease. And when we see the similar trend of the rust, we can see again on the 40th standard meteorological week only the disease was observed, but interestingly it was a reverse trend. You can see D3, that is third date of sowing, the maximum rust incidence was observed. So it was low in the first date of sowing, 35.2, 39.7, second date, and 47.4. And this is correlated with again the spore load. During the previous week, we can see the maximum temperature of 28.4, minimum temperature of 20.2, rainfall was 82.2 mm, 20 mm, 6 rainy days, morning and evening relative humidity varied from 96 to 88 respectively, and wind speed was 4.61 kilometer per hour. And you can see the graphical representation. The first one is early leaf spot. You can see the trend, increasing trend as compared to the rest of the 2 dates of sowing, wherein we can see the highest early leaf spot in the first date of sowing, the highest late leaf spot in the first date of sowing, and subsequent date is reduced in the second dates of sowing, which is again correlated with the temperature relative humidity, which I'm going to tell in the next slides. And the reverse trend was observed in the rust. We can see late planting has resulted in a heavy outbreak of the rust as compared to the first 2 dates of sowing. When coming to the correlation studies, it's very evident that we can see the cumulative rainfall in all the 3 dates of sowing. We can see the cumulative rainfall, cumulative rainy days, RH morning, and of course the RH2 in the evening. It has significantly contributed to the spore load, and that spore load has given rise to the maximum infection of early leaf spot. And when we consider the late leaf spot, here also only the cumulative rainfall of 0.950, 0.991, 0.98, which signifies 95%, 99%, and 98% respectively, along with the cumulative rainy days, they have contributed to the increase in the spore load and further leading to the leaf spot disease. And when it comes to the rust, here also the same cumulative rainfall and cumulative rainy days, and also temperature, minimum temperature, as well as these are the 3 major factors, and also evaporation. These 3 things or 4 factors have contributed for the outbreak of the rust disease in the groundnut. And we can see the 3 different dates of sowing plots, as well as this the spores trapped in the spore trap on the left hand side. And this is very important. The stepwise multiple regression is a very important phenomena when we are comparing all the 3. See, we should know that the first date of sowing, that is 22nd June 2017, cumulative rainy days have shown a significant positive regression with a coefficient R square value of 0.848. That means positive effect, and it alone contributes 84% to the disease development. Other factors did not contribute much to the disease. In the second date of sowing, 7th July 2017, cumulative rainy days and RH have shown significant positive regression coefficient with R square value of 0.972. That means it contributes 97% to the disease development. And in the third date of sowing, that is 25th July 2017, cumulative rainy days and RH, they have shown significant positive regression coefficient R square value of 0.987. It indicated that the cumulative rainy days and RH in evening have positive effect, and together they contribute 98% to the disease development. Other factors did not contribute much to the disease. For spore load, evaporation has shown significant negative regression coefficient with R scale of 1.465. This is with respect to early leaf spot. And coming to the late leaf spot, we can see DOS1, date of sowing 1, date of sowing 2, and date of sowing 3. When we focus there, the first date of sowing cumulative rainy days has shown a significant positive regression coefficient with R square value of 0.921. That is 92%. It has favored. In second dates of sowing, 7th July 2017, cumulative rainfall and wind speed have shown significant positive regression coefficient with R square value of 0.992. It indicated that the cumulative rainfall and wind speed here, please note here, wind speed, it has a positive effect on, together they contribute 99% to the disease development. And in the third date of sowing, 25th July 2017, minimum temperature, yes, here it is at minimum temperature and evaporation have shown 99.8%. And for a spore load in the late leaf spot, evaporation have shown significant negative regression coefficient with R square value of 0.592, which cannot be considered. Hence, apart from these, other factors for also contributing to the spore load present in the air. Regression equation disease index is 2.924. And when the rust is considered, the date of sowing cumulative rainfall shown a significant positive regression coefficient with R square value of 0.953. That means it contributes 95% to the disease development. In the second date of sowing, 7th July, as far as the rust is concerned, cumulative rainy days have shown significant positive regression coefficient with R square value of 0.965. The cumulative rainy days have positive effect, and alone it contributes to 96%. And in third date of sowing, the cumulative rainy days have shown significant positive regression coefficient R square value of 0.882. This means to say that they have shown a positive effect, and alone it contributes 88% to the disease development. And for spore load for rust, cumulative rainfall has shown a significant positive regression coefficient R square value as 0.935. That is, it contributed 93% to the spore load present in the air. Other factors did not contribute much to the disease development. So this is a very, very important as far as the disease prediction is concerned. And in the era of, we are talking about doubling the farmer's income. And this, the forewarning, if you give to the farmers or scientists working on different aspects of the diseases, if the forewarning is given to the farmers, they can very well take care of these fungal foliar diseases. Now coming to the third objective, that is management using chemicals and integrated approach, where it was used by using susceptible varieties JL24, treatments 11, 3 application, 2 sprays at 15 days interval. We can see the general view of the experimental site for management. We can see the different, all the 3 chemicals, T4, T5, T6, has significantly reduced the early leaf spot, late leaf spot, and rust in all the 3 diseases were reduced by this triazole. And my friend from Karnataka University has mentioned about the mechanism, its structure, about these triazoles. And see the treatment number 7, 8, 9. They are in combination with Pseudomonas fluorescens, one of the biological control agent. And in the era of organic, we have to also have to take care of the organics. Instead of taking 2 to 3 sprays with the chemicals alone, we have to take care of the crop as well as human health with the use of biopesticide like Pseudomonas fluorescens. They were found next best treatments after the chemicals. And because of the PGPR mechanism, we can see the difference in the plots. And we can see the treatments in untreated control as well as the tuberconazole. The plant, it was reflected in the plant height, number of nodules, and leaf area. We can see that because of PGPR mechanism, the highest plant height was observed, maximum number of nodules were observed, and also leaf area was also on par with the chemicals. And this is reflected in the graph. number of pods, 100 CD weight. Yield was also significantly superior in tebuconazole, propiconazole, and in the Pseudomonas fluorescens combination, they have given the next best treatments. And this is the most important. We can see the crop as well as the pods in different treatments. And of course, while using the biocontrol agents, natural enemies population will also increase. That will take care of all the ecology, and they are farmer friendly in nature. To conclude, maximum disease severity of fungal foliaritis was recorded in Dharwad. Dates of sowing revealed that early sowing escapes that rust, but the late sowing rust was usually comes faster than the early. Spore load for early leaf spot was highest at 38th meteorological week, and it was 40th meteorological week is for late leaf spot and rust. Correlation studies revealed that irrespective of the dates of sowing, cumulative rainfall and cumulative number of rainy days were significantly positive. And regression studies revealed that they are also effective. Plant growth and yield parameters were significantly enhanced because of reduction. PC ratio was also found maximum in chemicals treated plots. Thank you to all my supporters in the university, my students and staff, and the Secretary ISMPP for giving the opportunity. Thank you very much.
Okay, thank you, Dr. Gurudat Hegde. After long back, I'm meeting.
Thank you.
A nice presentation by your about the groundnut polarities, how this rainfall or the environmental factors affect on the diseases. Definitely, you have enlightened us with your presentation. Thank you once again for your good presentation. And I think timers, they have to mention the time about the presentation.
Sir, it is exactly 15 minutes, sir.
Okay, that's what.
Thank you.
I think he has covered very nicely, timely. And we shall go for the next speaker. I think we called first 4 on. Next speaker.
Sir, this is PP single is over, sir, now.
Next speaker is not there.
Next speaker is
The
not
fourth
there.
participant was there, no?
Yes, sir. One participant.
Once again, you call. If you're not mistaken, you please once again call.
No, he has communicated, sir. He is not going to participate.
Okay. Then it's good. Okay. We are. We had a 3 good presentations from 2 from Dharwad. That is Dr. Praveen from KUD Dharwad, and Dr. F.A. Mohiddin from Kashmir, and Dr. Gurudat Hegde from once again US Dharwad. We had a very good 3 presentation for Dr. P.P. Singhal Memorial Pesticide Industries Award 2020. Very good. We shall move on to the next Memorial Award, Dr. Guman Devi Verma Memorial Award. Let me call all the participants are there from central office. Dr. Mista, please.
No, sir. Yeah, Dr. Ms. Sneha Patil.
Yes, I am available.
Okay. Dr. Sangeeta.
Yes, he is there.
Sangeeta.
He was there, I think. I don't know.
Are you there?
Idro,
Okay,
I don't
she has
know.
logged in, but anyway,
Yes.
after her second presentation, and Shweta Singh,
Yes, sir, I'm there.
You're there. Okay.
Priya,
Yes, sir, I'm there.
Yeah. I think
then Durga
all of
Devi.
you know very.
Yes,
Durga
sir, I'm
Devi?
here.
Okay.
Yes,
So
sir,
it seems-
I'm here.
Okay, it seems all 5 presenters are present here.
Present.
Sir,
Yes.
It's over to chairman.
Okay, thank you once again. Hope all of you know the rules and regulations. For 50 total presentation is 15 minutes. So after 13 minutes, one bell will be given, and after the 2 minutes, long bell will be given. So you have to complete your presentation, all the presentation within the 15 minutes. Otherwise, the negative evaluation will be there. For that reason, all of you take care of your timing and present the things. No doubt. I think we have completed PP single like that only. You have to follow this women award, that is Dr. Guman Devi Verma award. May I request Dr. Sneha or Patil to present her presentation for the award?
Yes, sir.
And rapporteurs, I think kindly note down the rapporteurs, please. I am requesting the rapporteurs also, once again. And the co-chairman will be there. I think Dr. Kantraj is there. You are there?
Yes, sir. Yes. Here.
Okay. May I call Dr. Sneha or Patil for our presentation?
Yes, sir.
Yes, go ahead, please. Please.
I'm just sharing the screen. So is the screen visible?
It is visible. Once you start, the time
There is some echo problem, Mrs. Sneha.
Sir?
there is a echo is there.
Echo from your audio.
So is it okay now?
Okay,
Okay.
a little bit. Okay,
So is it okay now?
yes, yes, yes.
Okay, shall I start, sir? So good morning to everyone. Today I'm going to talk about the dark side of agricultural antibiotic streptocycline that is casted in tomato cultivation. So before beginning with it, I would like to give a brief gist about the importance of this topic. So as we know that tomato is the world's largest cultivated vegetable crop, and India ranks second in the global tomato production. There are around 7,500 cultivars of tomato available in India. And if we see that there are huge nutritional benefits of this tomato crop, it is a rich source of various vitamins like vitamin A, vitamin C, beta carotene, alpha carotene, lycopene, etc. And particularly, tomato has lycopene, which has added benefits as it is an antioxidant, and it helps in preventing cardiovascular diseases as well as cancer. Now, tomato crop is largely exploited for culinary purposes and in the processing industry. Like all other crops, tomato cultivation is also affected by various pests and diseases, among which the bacterial leaf spot of tomato caused by Xanthomonas campestris pathovesicatoria is the most devastating disease of the tomato, which causes around 80% reduction in productivity and can also cause around 10 to 20% annual yield loss, which may raise up to 80% under rare situations. So over the years, for management of this disease, antibiotics like streptomycin sulfate, tetracycline hydrochloride, and other copper-based compounds have been used. And these chemicals have been used repeatedly over the years, which has created various problems like development of streptomycin and tetracycline resistant bacterial strains, accumulation of these antibiotics on the soil particles affecting the quality of the soil and fertility of the soil. And also there has been reports of development of residual toxicity on the fruits. So to overcome these issues, and particularly the issue of resistant development, new antibiotics have been introduced, and one such antibiotic that has been introduced is the streptocycline, which is a combination product of streptomycin sulfate and tetracycline hydrochloride. So this was introduced by the Hindustan Antibiotic Private Limited, and it is widely exploited against various phytobacterial diseases in states of Maharashtra, Andhra Pradesh, and Tamil Nadu. So this experiment was framed with the view to validate whether these three problems associated with the use of other antibiotics stand true in case of the use of streptocycline as well. So for this purpose, the experiment was split up into three phases. In the first phase of experiment, we tracked the presence of streptocycline-resistant Xanthomonas campestris pathovesicatoria strains in Tamil Nadu, for which 20 different tomato growing hotspot regions were surveyed. And symptoms were observed on different plant parts. The symptoms on branches and the stems appeared to be round or oval, black or brown spots. On the fruits, the symptoms appeared as small brown to black spots and that were water soaked, and later on which become encrusted. And on the leaves, the symptoms appeared as small brown spots surrounded by a yellow halo which enlarged and covered the entire leaf as the disease progressed. Now, preliminary data regarding the percent disease incidence, prevalence, and severity for these 20 locations was collected, and it was observed that the incidence was around percent disease incidence was highest at Bagalur and Titipalayam. The prevalence of the disease was 100% in the regions of Bagalur and Titipalayam, and the disease severity on leaf was maximum at the region of Bagalur, and disease severity on fruit was maximum in the region of Titipalayam. Further, the infected fruits were collected from these 20 locations, and no variation in the symptoms were observed for all these locations. These fruits were used for isolation of the causal organism, and 24 bacterial strains were isolated from the infected fruits, and it was observed that all the 24 bacterial strains produced yellow mucoid colonies and scored positive for all the 7 biochemical tests that were carried out, thus giving a preliminary confirmation that the isolated bacterial strains belonged to the Xanthomonas group. Further, they were confirmed to be bacterial leaf spot causing Xanthomonas campestris pathovesicatoria through pathogenicity, as all these plants that were inoculated with the 24 strains produced symptoms similar to those observed under the field conditions. Next, we went in for molecular confirmation of the isolated bacterial strains using the universal primer targeting the 16S rRNA region, as well as the specific Xanthomonas campestris pathospecific primer, which was highly specific to the pathovesicatoria group. So both these experiments helped in confirming that the isolated 24 strains were Xanthomonas campestris pathovesicatoria. And further, the sequencing was done, and the sequences were submitted at NCBI Gene Bank, and accession numbers were derived. Next aim was to identify whether these strains are resistant to the antibiotic streptocycline or not, for which two methods were used. The first one was the minimum inhibition concentration assay, and the other one was the microbroad dilution assay. In case of the minimum inhibition concentration assay, the nutrient agar medium amended with 4 different concentration, that is 25, 50, 75, and 100 ppm of antibiotic streptocycline was used. And then the broth, bacterial broth consisting of 1 into 10 to the power 6 colony forming unit per ml bacterial population was evenly spread on the nutrient bed, and colonies were counted after 24 hours of inoculation. So the strains that failed to develop or failed to grow on the 50 ppm were considered as the sensitive strains, while the strains that had the ability to grow at the 100 ppm concentration were regarded as the moderately sensitive strains. So among these 24 strains, the strain S2, S10, S14, S17, and the strain S21 displayed growth at 100 ppm and thus were regarded as the moderately resistant strains. And this is the result of the microbroad dilution assay, in which a 9612 microtiter plate was used and 50 microliter of the antibiotic in four different concentrations and 50 microliter of the bacterial broth were inoculated, and the optical density at 620 nanometer was recorded after 12 hours of incubation. And again, similar results were observed, that is, the strain S2, S10, S14, 17, and 21 marked in red displayed higher growth at 100 ppm and thus were regarded as the moderately resistant strains, while the strains S4, 5, 6, 11, 13, 20, and 24 failed to grow at 100 ppm concentration and thus they were regarded as the sensitive strains. Now, these 5 moderately resistant strains were subjected to a higher concentration of 200, 300, 400, and 500 ppm concentration. And it was observed that among these 5 strains, the strain S2, S17, and S21 had the ability to grow at 500 ppm concentration and thus were regarded as the resistant strains. And among these 3 strains, the strain S21 had maximum number of colonies, and in case of microbroad dilution assay, had the highest growth at the 500 ppm and thus were regarded as the highly resistant strain. So overall, it was analyzed that there are 7 sensitive strains, 2 moderately resistant strains, 2 resistant strains, and 1 highly resistant strain present in Tamil Nadu for the antibiotic streptocycline. The next experiment targeted in analyzing whether these 24 strains were actually resistant to the core component of streptomycin sulfate also. So for that purpose, we have used the streptomycin gradient strip. So the nutrient bed amended with the bacterial broth was used and the strip was seated at the center of the plate, and the inhibition zone was recorded after 24 hours of incubation. So the larger the inhibition zone indicated that minimum concentration was required for inhibiting the bacteria, and thus that strain was regarded as the sensitive strain, while the smaller the inhibition zone referred to the presence of a requirement of a larger concentration of the antibiotic, thus regarding those strains as the resistant strains. So like the previous results, here also it was confirmed that the strains S2, S10, 14, 17, and 21 had the smallest inhibition zone and required the largest concentration of antibiotic and thus were regarded as the most resistant strains. The next phase of the experiment was to understand the influence of streptocycline on the soil quality and soil enzymes. So soil quality is reflected by various parameters, among which soil enzyme plays a predominant role as it catalyzes various reactions like the nutrient cycling of the soil, the degradation and transformation of plant microbes and plants and microbes, and degradation of the pollutants in the soil, carbon cycling of the soil. Thus, the soil enzymes act as a reflecting mirror for the soil quality and soil fertility. So owing to the importance of soil enzyme, the impact of streptocycline on 5 most important soil enzymes, that is soil dehydrogenase, catalase, soil urease, acid phosphatase, and soil beta-D-glucosidase was analyzed. For this experiment, the rhizosphere soil, tomato rhizosphere soil free from any antibiotics or any kind of chemicals for the last 5 years was used. The soil was treated with 250, 500, and 750 ppm concentration of streptocycline, and these concentrations were given in a set of 3 treatments. In the first set of treatment, these 3 concentrations of antibiotic were given single time. In the second set of treatment, these 3 concentrations were applied twice at an interval of 10 days. And in the third set of treatment, these 3 concentrations were given thrice at an interval of 10 days. And the enzyme activity were tested on the 0th and 7th day after the application of antibiotic streptocycline. The overall result for all the 5 enzymes revealed that as the concentration of antibiotic increased, there was a decline in the expression of the enzyme activity. The enzyme activity declined also for the second and third treatment, in which twice and thrice application were given for the 0th day as well, indicating that the previously accumulated molecules of the antibiotic hindered the expression of the enzymes. So which concluded that when an antibiotic is given for a prolonged period of time and irrationally, it leads to the accumulation of these antibiotics on the soil, which greatly reduce the effectivity of the soil enzyme as well as the fertility of the soil. So among these 5 enzymes, all the 5 enzymes showed a decline or negative correlation with the increasing in concentration and increase in number of applications of the antibiotics. Only variation among these 5 enzymes was the degree of decline of the enzymes. Otherwise, all of them showed a steep decline with the increase in concentration as well as increase in number of application of the enzyme of the antibiotics. The next aim was to identify whether there is a deposition or accumulation of these antibiotics on the edible portion of the plant, that is on the tomato fruits. For this purpose, we aimed in understanding whether both the components, that is streptomycin sulfate and tetracycline hydrochloride, which together form the streptocycline antibiotic, are depositing separately on the tomato fruits. So for this purpose, high performance liquid chromatography was used, and a similar kind of experimental setup was used, that is 250, 500, 750 ppm was applied on the tomato plant, and 3 different set of treatments, that is single application, double application, and triple application. And following the application, after 45 days of application, the fruits were harvested and subjected to the HPLC analysis. So 6 different parameters were used for understanding or validating the experiment and the instrumentation. So among the parameters included recovery percentage, limit of detection, limit of quantification, intercept slope, and the correlation coefficient. So the recovery percentage refers to the concentration of antibiotic that can be recovered from an instrument when a known concentration is injected into the instrument. So the recovery percentage was around 86.67 for streptomycin sulfate and was 91% for tetracycline hydrochloride. The limit of detection refers to the minimum concentration that can be detected by the instrument for a given method, and it was 6 ppb for streptomycin sulfate and 5 ppb for tetracycline hydrochloride. The limit of quantification refers to the minimum quantity that can be quantified by the instrument for a given method, which was 18 ppb for streptomycin sulfate and 17 ppb for tetracycline hydrochloride. The correlation coefficient was 0.99% for both the chemicals, thus validating the instrument and the method. Now, this is the chromatogram for the 5 different standards, 5 different concentration of streptomycin sulfate standard of 99% purity that was injected into the instrument at 50 ppb, 100 ppb, 150, 200, and 250 ppb, and a spike of 100 ppb was given, which helped in the analysis of the R-square value, the intercept and slope value, and the recovery percentage through the multi-point calibration curve. And this is the liquid chromatogram of streptomycin sulfate, which was derived from the tomato fruits that were given different treatments of different concentration and different number of applications. The retention time for streptomycin sulfate was 1.2 minutes, and the peak area was used for calculation of the final concentration that accumulated on the tomato fruits. So the minimum concentration of streptomycin sulfate that accumulated on the tomato fruits was 26.32 microgram per kg of the fruit, and the highest one was 103.97 microgram per kg of the fruit. And it was observed that as the concentration of antibiotic increased and as the number of application of the antibiotic increased, there was an increase in the concentration of accumulating antibiotic on the tomato fruits as well. Likewise, the same set of experiment was repeated for tetracycline hydrochloride. The multi-point calibration curve was analyzed using the 5 concentration of standards, that is 50, 100, 150, 200, and 250 ppb, and a spike of 100 ppb. And the retention time for tetracycline hydrochloride was 10 minutes. And these are the peak areas for all the samples that were tested for different concentrations and different number of applications. Here also it was observed that as the concentration of antibiotic increased and the number of application increased, there was an increase in the accumulation of the antibiotic on the final produce, that is on the tomato fruits. In case of tetracycline, the minimum residual toxicity was 42.83 microgram, while the largest concentration of accumulation was 116.86 microgram per kg of the fruit. And the cumulative effect of both the streptomycin and tetracycline hydrochloride together also were within the level of acceptance for human consumption, thus making it so far fit for human consumption. But if this level goes on increasing, if there is a more prolonged exposure of these fruits to the antibiotics, there will be a higher concentration of accumulation on the fruits, which will surpass the level of accepted level for human consumption, making it unfit for human consumption and unfit for export also, thereby reducing the GDP of the country. Thus, it is essential that the antibiotics, when they are used, they should be used in a proper proportion and under proper supervision, so that there is a limited residual toxicity which is within the range of human consumption. So far from this experiment, we have understood that there is the presence of streptocycline resistant strains prevalent in Tamil Nadu, and the overuse of antibiotic has led to the adverse effects on the soil enzyme and also led to development of residual toxicity on the fruits. So now, as plant pathologists, it becomes imperative on us to develop alternate strategies so that the use of streptocycline can be gradually declined. So as an alternate strategy, when we are developing, we should keep in mind that this strategy should be effective against the already existing streptocycline resistant bacterial strains, and they should also be environment friendly. So keeping all these parameters in mind, we opted in for the use of plant extracts. So we have used 25 normally available plant extracts, that is neem, vinca rosea, eucalyptus, vimmu, lemongrass, thyme, sweet basil, adathoda vesica, coleus castor, Herpelia cupenoides, mint, lantana camara, Parthenium hysterophorus, papaya, ashwagandha, nochi leaf, nochi flower, pungam, ginger, garlic, onion, turmeric, and bael, and tested their antibacterial activity, as well as we have tested the synergistic effect with the antibiotics. So for evaluating the antibacterial activity, the aqueous extract of 1, 3, and 5% of these plant extracts was used, and it was observed that neem, eucalyptus, vimmu, lemongrass, Herpelia cupenoides, lantana camara, Parthenium hysterophorus, tulsi, ginger, garlic, and turmeric had the ability to inhibit the bacteria at 1% aqueous extract concentration itself.
Hello. This
Yes,
is to
sir.
interrupt Mr. Sneha Patel. You have already exceeded time limit.
So just 2 more minutes.
No. See, please conclude. Please conclude it within 1 minute. As per the regulations, you have to finish within 15 minutes.
So these 11 plant extracts were effective at 1% concentration, among which garlic had an effect that was larger than the antibiotic control itself at the 1% concentration itself. So garlic produced 40 mm inhibition zone, which was larger than that of the antibiotic control. Next, the synergistic effect was analyzed, which again showed that all of the 11 effective plant extracts showed synergism with the antibiotics, and among this, garlic had an inhibition that was double the time that of the antibiotic control, thus confirming that garlic can be widely exploited for management of this disease and can be used as an alternate for the antibiotic control. So with this, I would like to conclude that although antibiotics are the need of the hour as they help in management of the disease, but they have produced drawbacks that can be affecting the crop cultivation in the long run. So as a result of which, we should opt in for other alternatives like using of plant extracts, which have added benefits that they are environment friendly, they are non-toxic, they do not form any residues, and thus they can be widely used for the management of disease so that we can reduce the effect on other environment parameters. Thank you.
So she has taken 2 minutes extra. Hello.
Okay.
Hello.
Thank you, Dr. Sneha Patil, for your nice presentation.
Thank you, sir.
Okay. May I call next participant, Dr. Sangeeta?
Yes, sir.
Yes,
Yes,
madam.
sir.
Please go ahead.
Yes, sir.
For your presentation.
Oh, sir, host disabled participant screen sharing.
Madam, enabled, please.
Can I? Yeah. Yeah, can I, sir? Good morning, everyone. Good morning, the organizing secretary, chairman, and the co-chairman and all the delegates. I'm here to present my topic on exploring microbial endophytes as potential antagonists against Colletotrichum gliosporoides causing anthracnose of mango. As we all know, mango is one of the most important fruit crop which is grown widely in all the tropical and subtropical areas of the world, and is one fruit which is in most demand compared to any other fruit. Now, when we look into the production of mango, more than 90 countries produce mango, wherein Asia accounts to about 73% of the mango production. And when we look into the Indian scenario, India is accounting for up to 52% of the global production of mango. When look at the problems with respect to mango cultivation, mango is being infected by various pests and diseases. Among them, the diseases form a major part. Among the diseases, we have post-harvest decay or spoilage, which accounts to about 25 to 40% of spoilage in India. Now, coming to the various post-harvest diseases which have been reported on mango, there are a few of them. Among them, anthracnose, which is caused by Colostotrichum gliosporidis, is the most important post-harvest disease which have been accepted globally. The disease is present both as a pre-harvest.
Madam, please be loud.
Yeah. Sir, can you hear me, sir? The disease.
Yes, madam. Yes.
Yeah. The disease is present both as a pre-harvest and a post-harvest disease, but the post-harvest anthracnose is the one which is causing severe losses both qualitatively as well as quantitatively, which is accounting of up to 30 to 60% loss in India. Now, to manage this anthracnose, various approaches have been used by different people, but we wanted to use an eco-friendly approach. So we wanted to concentrate our work on isolation of endophytes, which are ubiquitous microorganisms, which are present in almost all the parts of the plant. And the habitats vary from terrestrial to aquatic, and major endophytes have been reported are fungi and bacteria, though an algae and actinomyces are isolated. When we look into the various different crops which have been used for extracting endophytes which exhibit antimicrobial activity, the major ones have been the medicinal plants, which have contributed to up to 35%, followed by other crop species contributing to 29%. Now, endophytes have been able to give a protection against both biotic as well as abiotic stresses. Many workers have reported the isolation of various endophytic fungi exhibiting antimicrobial activity against many of the pathogens causing diseases in major crops. With this background, we have taken our objective to isolate fungal and bacterial endophytes from different varieties of mango and to prospect their antagonistic activity against the Colostotrichum gliosporidis, which is causing anthracnose in mango. So first, primarily, we started with the isolation of the pathogen from the infected fruit sample, and got the culture and the conidia, and also we proved the pathogenicity. Next, for the isolation of endophytes, we collected samples, the leaf and stem samples from 10 different varieties, out of which 3 we got from our own campus, whereas the other 6 varieties we collected from IHR Esarghatta, Bangalore, and some of these varieties have been reported to be moderately susceptible and are more than about 10-year-old trees from which we collected the samples. Now, the protocol which we used for isolation was the standard protocol for isolation of endophytic fungi, wherein surface sterilizations were done with ethanol and sodium hypochlorite. And after the last wash with sterile distilled water, an aliquot was plated on dextrose, the media, to confirm the sterility. If any contamination was seen in this particular plate, the entire experiment was repeated again. When we did an isolation, we got a wide range of endophytic fungi from the leaf as well as the stem sample, whereas the maximum number we got was from the Alfonso variety, 4 isolates from the leaf sample and 5 isolates from the stem sample. And but Neelam variety, we did not get any from the leaf, whereas we got 4 from the stem sample. So further, we studied the morphological characteristics of these isolates. When we studied the morphological characteristics of these different isolates, we found that most of the isolates from the Alfonso leaf variety were all septate and colored, whereas 2 varieties which I have highlighted here, one was for slow growing and one was the fast growing. From the stem isolates, all the isolates were aseptate and hyaline. This is the picture showing the different isolates with the mycelial characteristics. Then from the Totapuri variety, the leaf isolates were all, 2 were aseptate, whereas 2 were septate. And among the stem isolates, 1 showed the aseptate hyaline mycelia. This is the picture showing the different color characteristics. From the different other varieties, there was one isolate which was isolated from the stem of Neelam, which showed aseptate hyaline. Further, we studied characteristics. One particular isolate from Villard showed did not form any spore, whereas it was colored and showed septate hyphae. Then based on this pictures, we further went for studying the antagonistic activity of these different isolates following the dual plate culture. From these results, we were able to find that one leaf isolate from Alfonso inhibited the pathogen up to 72.32% and 71.11% from the stem isolate. This is the picture showing the inhibition or activity of the stem isolate and the leaf isolate. Further, from the Totapuri, we had got another isolate which was from the stem, which was designated as E, which showed about 55.92% inhibition. Other varieties like Neelam, the isolates did not show much inhibition, which was less than 50%. And even the isolate from Kisan Bhog showed about 60% inhibition. One major isolate which we got from the Villard stem isolate, which was designated as EFWDS, showed about 83.70%. So based on these studies, we were able to square down or come down to 3 isolates which were showing maximum inhibition. Among this, the WDS from the stem isolate was showing maximum inhibition, and it was also a fast growing fungi. So we went for characterization of this particular isolate, wherein we went for PCR analysis with the genomic DNA using the universal primers, and we got a 700 base pair product, which was further subjected to homology studies. And based on the homology study, the isolate was identified as Diaporthes citri. Further, when we went for the isolation of the endophytic bacteria, again using the same protocol which was followed for fungi, we were able to get about 10 isolates, just one isolate from each of the 10 varieties from the leaf as well as the stem. And when we did morphological study, one isolate from the Anfas showed rod shape and gave a gram-negative reaction. And from this, these are the bacterial isolates isolated from different stem and leaf. Then further, we went for the dual culture assay. From the dual culture assay, from the leaf isolates, we got one from Anfas, which showed about 56.67% inhibition of the pathogen. And one more isolate from the Kisan Bhog, what showed 53% inhibition of the pathogen, whereas from Alfonso, the stem tissue bacterial isolate showed about 54.74% inhibition of the pathogen. So basically, we got again 3 endophytic bacteria which was showing inhibition of the pathogen, but the inhibition by the endophytic bacteria were not as much as compared to the endophytic fungi. Maximum inhibition which we got with respect to endophytic bacteria were only 56.67% from the leaf isolate of the Anfas variety. So based on these studies, we further went in for the identification of the different isolates of the bacteria, and we went for the genome isolation, genomic DNA isolation and sequencing using the 16S rRNA gene. And when it was amplified, we got a band of 1500 base pair which was observed on the PCR gel. And further, when it was subjected to homology studies and sequence alignment from the results of the BLAST sequence, the particular bacteria was identified as Pseudomonas plicoglossicida strain. So based on this, we have come to the conclusion that we were able to get one endophytic fungi and one bacterial strain which was showing good inhibition of the pathogen, but there was a lot other problems associated with these endophytic fungi. So further, now we have to test for the efficacy of these isolated endophytes for their inhibition of the pathogen on the infected fruit. Whether we were planning to treat the infected fruits with the following with the dripping of this fruit in this endophytic fungi or bacterial solution, and then study what would be the microbiome effect of these particular endophytes on the fruit, what would happen to the microbiome which is already present on the fruit as well as on the tree in the field conditions. So I thank the organizers for giving me an opportunity to present my work. Thank you.
Sir, Madam has taken 9 minutes 52 seconds to finish. Sir, Chairman, sir.
Okay, presentation about. Yes, yes, yes.
Sir,
I'm
please.
thanking Dr. Sangeeta Madam
Yeah, thank you, sir.
for enriching our knowledge about endophytic fungi and bacteria against Coleoptera sporoides. And thank you for your nice presentation. And may I call Dr. Shweta Singh for her presentation? Yes, madam, please go ahead. Please, please.
Shall I begin, sir?
Yes, yes, madam, go ahead, please.
Respected Chairman, Co-Chairman of the session, Honorable Jury, Senior Delegates and colleagues from different parts of the globe, good morning to all. Sir, I'll be presenting my work on characterization of indigenous wild split-gilled mushroom and development of cultivation technology in Northeast India. This is part of work done by our team at Sikkim when I was posted at ICR Research Complex for NEH region. Yesterday, we all got a chance to listen to Dr. Krishnamurthy and his talk on mushrooms, and we are pretty well aware that mushrooms, they have become an integral part of our diet owing to their flavor, nutrition, and medicinal properties. So in that context, I'll be presenting my work. So almost 2,000 edible species of mushrooms are known to mankind, of which almost 50 species are only consumed commonly, of which button, oyster, paddy straw, milky, and shiitake are very commonly known to all of us. But this split-gilled mushroom schizophyllum commune is one which is very uncommon to people, and also that it is uncommonly worked upon. Not much research has been also done on this. However, this is a very nutritionally and medicinally rich mushroom. Taxonomically, this mushroom falls under the phylum Basidiomycota and order Schizophylles. And the schizophylline has a characteristic property that all the genus in this has split gills. So this term schizophylum implies that split gills, and commune means common, that it is this common property is shared across the world by this particular fungus. And the schizophylum commune grows commonly on the decaying trees, and whenever there is a dry spell, so we can collect these mushrooms naturally from them. As we all can see in the picture, the above part, the upper part is like just another mushroom, white bracket mushroom, but beneath the cap there are radial gill-like folds, each of which is centrally split, and hence it is called as split gill. So this is a clever adaptation of this mushroom to the changing environment, and it is consumed across the globe. And in India, it is a very flavored dish in the northeastern community, especially by the Mizo, Sikkimese, and the Manipuri people. So this has a very alternate food source in the future. Also, it can be used for industrial applications by tapping its nutraceutical property, which is rendered by a glucan carbohydrate, schizophyllan, and the immunomodulatory antifungal and antiviral activities of this is higher than any other glucan mushroom. This study was designed with objectives to characterize it on important parameters like morphological, anatomical, cultural, and molecular basis, and to get an insight into its features. Also, we propose here a technology, cultivation technology of this mushroom on different agricultural residues which are of abundance. So, as we all can see here, this mushroom fruiting body the bark of Aloes nepalensis and Albizia chinensis, which is commonly found in Sikkim. And these basidiocarps were collected from the research farm of ICR Sikkim. So on this bark, we can see that the basidiocarps are arranged in a tier system, one over the other. And this mushroom gets its nutrition from the by consuming lignin from the timber, and it leaves behind the cellulose. So it is also known to called wood rotting or white rot of woods. This mushroom has a typical characteristic feature or adaptation that it can rehydrate itself and also dehydrate itself. When there is abundant moisture, it will be rehydrated. And when there is a lack of moisture or dry spell, it gets dehydrated, because of which this can survive several cycles and it can survive in any continent of the earth. So the first basic step was to collect spore print of this fungus, which is a traditional diagnostic method where the spore color and features help in identifying it. So it is done, here we did it on black paper and transparency sheet, used it for microscopic studies as described by Kumar et al. And Atri et al. The morphological characterization was done. So here we can see the dorsal surface, the pileus, is flabelliform or fan-like in shape and spongy and dense structure. This pileus, when it is young, it is white in color and it becomes brownish upon maturity. Here in the picture is a mature pileus, so it is pretty brown in color. And also the whitish tinge which we can see on the upper surface is because of presence of white hair on the surface. So this cap is, the length of this cap is usually 1 to 4 centimeter across, and it is about 0.3 centimeters. In this case, it is mostly a stipeless mushroom. If you look on the ventral surface, here there are two pictures. One is the moist basidiocarp, which is completely hydrated, and the lower one is the shriveled basidiocarp, which is actually dehydrated and curled inwards. But in both the cases, we can see longitudinally running gills, which are split from the center. All these gills, they come and close at the end. The end part is the hymenophore, or the fertile part from where the spores are actually produced. And in the shriveled basidiocarp, this curling is also a protective strategy of this mushroom, because by curling, they try to protect their fertile part so that when they get moisture, they can again produce spores. The cultural characterization was done by growing it, by obtaining a pure culture of this fruiting body on PDA media, as proposed by Chang and Miles. And this, we could see the complete white woolly mycelial growth on the plate after 5 to 6 days, followed by formation of this characteristic bump in the center. We could get this upon 9th or 8th or 9th day of the incubation. The reverse side was yellow-brown in color, and this pure culture has been submitted to ICR, Directorate of Mushroom Research, Solon, and we got accession by DMRO 1184. The next is the submerged culture. We also attempted this submerged culture in potato dextrose broth, so that we can see how it grows on that. So initially, there was 5 to 6 days, we could see white mycelial growth on the broth. After that, small protuberances, aerial protuberances started growing on it. And what we can see here in the picture is finger-like projections of this basidiocarp, which actually we get it on 20th day. So these fingers measured around 4 centimeters in length and were about 0.1 to 0.3 mm in width. The anatomically also we try to characterize it by using the spore print and the mycelia from the pure culture. Here we are showing the trama of the mushroom, which is in form of interwoven longitudinal hyphae, and it is multistratus in nature. And when it is, this hyphae is usually compact, but when it penetrates out of the pileal region, it gives a hairy appearance. This is the heteromerous trama. Heteromerous means there are 2 to 3 different types of hyphae inside that. So within this heteromerous trama, we can see spherocysts inside, and with a lot of air lacuna. These spherocysts are actually globose or cylindrical in shape. At the same time, at one end, we can see young basidia formation, which is arising from the subhymen layer. Then there is cystidia, which is intermingled with sterile hyphae. Next is this hyphae of the mushroom. Usually, Schizophilum commune is monomitic in nature. That means it has only one kind of hyphae. Very rarely we get to see dimetic also. So in this case, the monomitic, it is monomitic and it has only generative hyphae. So in these 3 pictures, we can see 3 different behaviors of generative hyphae, where in the first picture, it is loosely arranged. in the last picture it is very compact, as well as in the but in the middle picture it is branched, and also we can see septations inside this hyaline hypha. Coming to this generative hyphae, we were also able to get dikaryon formation inside the hypha, clamp connections, and also the dikaryon formation in this mushroom. The basidiospores were cylindrical and hyaline, and they were not at all amyloid. So, and at the distal end, there was a small tuberance, which is called as hilum, and it represents the conjunction with the stigma. And here there are two different kinds of basidiospores. The lighter ones are the ungerminated basidiospores, while the darker ones are germinating basidiospores, which can be differentiated because the ungerminated ones are lighter in texture, which is probably because the mitochondria is disorganized, and also carbohydrate is the reserve food here. While in the germinating basidiospores, the mitochondria gets very well organized and becomes condensed, and here the lipid serves as the reserve. Coming to the molecular characterization, the molecular characterization was done by sequencing the ITS region of the DNA, and here we use the ITS1 and ITS4 primers. The DNA was isolated as per the protocol described by Arvin et al. 2013, and the gene blast was used for identification of mushroom. The primer sequences and the thermocycler conditions are mentioned here. So the size of amplicon, what we got from the PCR was around 650 base pairs, and upon sequencing, the sequence was matched in NCBI blast, where it showed 100% similarity with Schizophilum commune gene bank accessions, and the native strain Schizophilum commune was also submitted in the database, where we could obtain the accession number MW504729. We also constructed a phylogenetic tree with 15 other gene bank submissions, and what we got was that almost all the 15 strains, 15 accessions showed similarity with the native strain of Schizophilum commune. There was it was completely similar, so we had to go for multiple sequence alignment. And if we look here very closely, the 6th base pair of G and the 16th position of A, these are the only two places where we find variation, very slight variation. Otherwise, all the sequences were completely similar. Coming to the chemical characterization part, it was very important to do this because we wanted to analyze the nutrient composition of this native strain of Schizophilum commune. So, and at the same time, we have done domestication of this strain also. So here we have one cultivated and the other one is wild, and with an objective to see how rich it is nutritionally with other commonly available mushrooms. This study was done. So here what we could see was there was the domestication increased the protein percentage, carbohydrate percentage, and the energy of the Schizophilum commune as compared to the wild one, and also that the Schizophilum commune is almost equal or slightly lesser than oyster or white button mushroom, which we consume commonly. So this particular mushroom has huge scope in our diet. Then another one was mineral composition, which was done using AAS and ICP-AES. The study was done at our headquarters in Barapani. So the Schizophilum commune is the nitrogen was 3.25% and potassium 1.12%, and the other iron, zinc, copper, and manganese were also estimated. The most important part was the cultivation technology. And the first step in the cultivation technology was to develop the spawn, which was prepared on paddy grain substrate, which is very easily available in Northeast India. And the techniques were followed as standardized by ICR-DMR. So we could get spawn run in 13 to 15 days, after which a small characteristic bump was observed on this. And the very typical characteristic of this spawn was that we could get a very disagreeable tarty smell out of it. Substrate requirement and substrate standardization is the most important part of mushroom cultivation. So recently we have all seen that agricultural residue has become big issue because there is a problem to dispose them of. And literally, India is producing almost 350 million tons of agricultural residue every year. And in Northeast India, we have paddy straw, wheat straw, buckwheat straw, and sawdust of different hardwood trees, which can actually be utilized for mushroom cultivation. So here, on the basis of availability and economics, we selected wheat bran, sawdust, wheat straw, paddy straw, and different combinations. And these were all supplemented with gypsum and lime as per the recommendations. And the performance of treatment was evaluated on the basis of spawn run, days for harvesting, fresh and dry weight, and the biological efficiency. So out of these, we made 6 combinations from which the paddy straw and wheat bran in a ratio of 2:1 supplemented with lime and gypsum performed the best by giving a yield of 117.51 grams, fresh yield and dry weight of 30.57 grams, and a biological efficiency of 23.4, which was followed by wheat straw and wheat bran in a ratio of 2:1. These are the pictures of the fruiting of schizophyllum on different substrates, and we can see that the fruiting on paddy straw is very prolific as compared to sawdust, and because we have used wheat bran, the fruit size and fruiting intensity is also higher. So with this, I would like to conclude that in this study, we have tried to characterize the indigenous strain of nutritionally rich mushroom, and the nutrient profiling promises that it can be a potential candidate for future food source, because the coming generation is very health conscious, and it will also address the issue of huge volumes of agricultural residue by transforming it into some marketable biomass of market value. Plus, we are also giving a technology for commercial cultivation of this mushroom using paddy straw and wheat bran. And this technology will help this mushroom, which is undervalued mushroom, to come up and compete with the commonly available mushrooms like button, oyster, or ear mushroom, or lion's mane mushroom. And at the same time, we have also come out that the domestication of this wild mushroom has enhanced its fruit size, given better carbohydrate, protein, and energy levels. And the submerged culture on liquid media can be utilized on large scale for enhancing the production and isolation of schizophyllan, which is a pharmacologically active polysaccharide, and it will be of huge industrial demand. The future areas that can be worked upon for this is that, because this is just a basic work, and I hope, to the best of my knowledge, this is first time ever in India that cultivation technology for schizophyllan commune has been taken up. So maybe in future more indigenous strains can be targeted, and more of better performing strains can be done. And then industrial level tapping of schizophyllan can be done, and more awareness and capacity building activities can be taken up for this mushroom. I would like to place on record my gratitude to the scientists and staff of my previous institute, ICR Research Complex, for NEH region. Hello.
Sorry for interruption, Shweta. Your time, you have already exceeded 2.5 minutes.
Okay, sir.
Wind up.
Okay. Thank you, sir. That's what I'll conclude, sir. Thank you so much.
Okay, Dr. Shweta Singh, thank you for your nice presentation about the wild split guild mushroom. Nowadays, everyone is very cautious about food.
But just please note that she has taken 2.5 minutes extra.
I think may I call Dr. Priya Iyer for her presentation. May I call Dr. Priya Iyer?
Yes, sir, I'm here.
Oh, you get. Yes, go ahead.
Priya, please on your.
तो फाइलम कम्यून कितना टाइम लगेगा किसमें चल रहा है सिस्टम देख लें 2-4 और नहीं
I request others sir to get mute.
Sir, is it visible and audible?
Yes, it is audible.
Okay. So shall I start now?
Okay, you can start now. No problem.
Good morning and welcome. Today I'm here to present on the topic development of advanced diagnostic tools and post-transcriptome analysis of dual CNDV associated with yellow mosaic disease of Ridgegard. As you know, Ridgegard, scientifically known as Lupha acutangula, belongs to the family Cucurbitaceae with the chromosome number 2n is equal to 26. It's basically originated in India, popular among South India. Alone, Karnataka contributes about 5,408 hectare area with a production of 56,726 metric tons with a productivity of 10.49 tons per hectare. Though it is affected by so many pests and diseases, the majority are powdery mildew and downy mildew, cholera rot and pythium. Apart from that, during 2013 and 15, the North Karnataka parts, mainly Belgavi, Bagalkot, Vijaypur, Dharwad, Haveri and Gadag, affected by the new disease that is yellow mosaic disease, has been reported with the symptoms of light chlorotic specks on the leaves with the yellow mosaic symptoms. In the later course, cupping of the leaves, and there is a huge reduction in the growth as well as in the yield. So during 2017, Patil and his co-workers characterized the disease based on symptomatology, transmission and molecular methods and proved the disease was caused by strain of tomato leaf curl New Delhi virus. The crops may often affected more than one or more pests or disease or strains of the same pathogens. This leads to the crop loss due to the untailoring of the specific treatments for the management of the same. So there is a need to identify the disease as soon as possible to tailor the specific treatments to minimize the crop loss. Even the breeders, the symptoms alone may confuse the breeders to breed the specific pathogens against the resistance. So this needs a proper diagnostic tools. With this concept, we developed a serological and molecular diagnostic tools for the detection of virus. We standardize the serological tools and molecular tools. Under serological tools, ELISA, DIBA, Western blot, and immunocapture assays were standardized. Under molecular tools, real-time PCR, LAMP, and RP assays were standardized. We'll go one by one. First, to standardize serological tools, I purified the recombinant protein by using 12 NDV code protein and produced the polyclonal antibody using recombinant DNA technology. For this, the code protein gene was designed with the BAM-H1 and ND1 restriction sites at 5' end of the forward primer and HIND3 at the 5' end of the reverse primer. And the code protein was amplified using the infected leaf sample and subjected for cloning and in PBS vector. These clones were confirmed by using both PCR as well as the restriction digestion method by using BAM-H1 and HIND3 restriction sites. After confirming the cloning, these clones were subjected for subcloning for expression purpose in PET vector with the ND1 and HIND3 restriction sites. These clones were also confirmed by using PCR as well as restriction enzyme method, and also by sequencing. So sequencing using NCBS data analysis. Before going for the expression, we studied the physicochemical characteristics of code protein to know the characters of protein extracted from the tomato leaf curl New Delhi virus protein. So this concludes the molecular weight of 2.9 kDa and with the isoelectrical point of 10.15. This indicates the protein is in the basic form, and the improbability of expression in inclusion body was 0.3%. This indicates the protein is ability to form the inclusion bodies up to 50%. So with the background knowledge, we isolated the code protein in PET vector, PET22B vector, and purified the protein by using different methods, 20 degrees centigrade, 30 degrees centigrade, and 37 degrees centigrade. This clearly indicates the protein is ability to form the inclusion bodies at higher temperature. So we selected the 20 degrees Celsius for the isolation, so for the extraction of protein. After this, the protein was extracted by using the detergent, that is 0.5% Triton X, because it is ability to form the inclusion bodies is more. That's why we used the 0.5% Triton X and we purified the protein. And this was confirmed by using the Western blot analysis using anti-his-tag antibody. The protein was immunized for 12 weeks in a rabbit, and third booster dose was analyzed for the reactivity assay. This concluded that the dilution of 1:40,000 is up to 1:40,000. It can give the positive reaction against the 2-LCNDV recombinant protein. So this was again confirmed by using the Western blot analysis. The antibody thus produced was used for the detection of 2-LCNDV by using plant sap. This concludes that is up to 1:20,000 dilution. The antibody is able to react with the host 2-LCNDV infected plants, and thus it also used for the further studies like dot immunobinding assay and as well as Western blot. Coming to molecular methods, we standardized the real-time PCR assay to detect the 2-LCNDV in RichGuard. For this, we designed the primers against code protein. This is the primer and the primer sites. For this, initially we subjected the plants for the artificial inoculation with the different time intervals and collected the leaf samples for the detection as well as the detection. For this, we plotted the standard graph by using PBS vector, which was cloned earlier using the code protein gene. And you can see the difference here. The code in normal PCR, normal PCR, it is able to detect the PCR product up to 6 hours after inoculation. But in case of real-time PCR, it can able to detect the pathogen in a short interval of time up to 3 hours after inoculation with the estimated copy number of 2.74 into 10 raised to 2 with the CT value 26.34. Even you can detect the virus requires only 3 hours to infect the plant. Second one is loop mediated isothermal amplification. This method is very convenient than normal PCR method. It's not required any sophisticated lab. Just by using isothermal condition, maintaining by hot water bath, we can detect the pathogens. This utilizes 3 set of primers: forward outer primer, backward outer primer, and loop primers to detect the pathogen in presence of BST polymerase enzyme. So we standardized this technique by different methods, that is incubation temperature, reaction time, outer and inner primer concentration. So with regards to incubation temperature, the maximum temperature maintained here is 65 degrees centigrade, and reaction time, so it can able to detect the pathogen within a 15 minutes of time. Outer temperature, outer and inner primer concentration, that is up to 1:1. Regarding sensitivity assay, it can able to detect the pathogens up to 1:10 to the power minus 10 dilution than the normal PCR. It can able to detect only 10 raised to minus 7. So this indicates the LAMP assay is very sensitive. It can detect the pathogens at very low concentration than normal PCR. Here we use the DNA intercalating dyes to avoid the hazardous chemicals using in the agar gel electrophoresis. It can able to, that is ETBR, very PCR, H1B and thiazole orange were used. All the 4 dyes are used in detection to avoid the hazardous chemicals using in agar gel. Next one is recombinase polymerase amplification. This is also a very convenient method as like isothermal amplification. Here there are 3 set of enzymes: recombinase, single stranded DNA polymerase, then displacing DNA polymerase. All the 3 set of enzymes were used for the polymerase assay to give the positive reaction. Here we designed the primers against the against the primers, then this gives the positive amplification of 214 nucleotide size. So now the question is to minimize the time consumed by the detection. So we cut down the DNA extraction method by using immunocapture assays. Here, this is a combination of both serology as well as molecular techniques. There is no need to go for DNA extraction. Directly, we capture the protein or capture the virals from by using antibody, then we'll go for the molecular techniques. These methods almost cut down the DNA extraction method to minimize the detection time. So this is the result. You can see the PCR as well as in lamp. First, first the plant sap will be trapped by the antibody generated by polyclonal antibody production, then utilized for the PCR and lamp. This almost take around 3 to 4 hours to detect the pathogens. Again, here also we use the DNA intercalating dyes. This gives the positive reaction in all the reactions. So this diagnostic tools we validated by collecting the different infected samples by all over the South India. In all the techniques are able to detect the plant pathogens in all the assays. This is the pictorial diagram. To know the cross reactivity assay of the antibody, we collected the different begovirus infected plants from all different crops, from watermelon, cucumber, pumpkin, bitter gourd, bottle gourd, snake gourd, and chili, capsicum, poppy, and beans. These are the primers used to detect the begovirus. Core protein primers were used. Here you can see the all the plant sample shows the presence of begovirus, and the same sample subjected for the TULSND specific primers. Only capsicum, chili, copy, and French bean are absent in this assay. So further this subjected for lamp assay. Here also we got the same result. Then subjected for the polyclonal reacting with a polyclonal antibody. All the cucurbitaceous plants are shown very positive reaction with the antibody except chili, capsicum, coffee, and beans. This is a pictorial diagram. So this the same used for the immunocapture assays also. Here also we got the same results as like PCR and ELISA method. To know why it is reacting with the coat protein polyclonal antibody produced by the TULSNDB. So we downloaded the different sequences available in the NCBS and subjected for the sequence identity. This reveals the tomato leaf curl palampu virus and squash leaf curl China virus have more sequence identity with the tomato leaf curl than chili leaf curl virus and mung bean leaf curl virus. To confirm this, we subjected the samples to DNA sequence using DNA primers and get sequenced. The results also as compared with the previous cluster W analysis, that is it gives the positive, it same as in case of cluster W analysis. So later we screened about near 150 varieties and hybrids for the resistance, but unfortunately we didn't able to get any resistance source. That's why we went for transcriptome analysis to know the transcriptional changes between healthy and tomato leaf curl infected plant samples. So these are the plant samples. So artificially we inoculated the plants for the by vector and confirmed the presence of TULSNDB by using TULSNDB coat protein specific primers using PCR and subjected for transcriptome analysis. Here in transcriptome analysis we got the around 2 crore reads. After that we filter the all the reads, and the number of reads we are used up to 1 lakh reads. The transcript generated here is in healthy, there are around 64,000 to 85,000 transcripts were generated in healthy, and in infected 69 to 78,515 transcripts were generated. These generated transcripts were used for differential gene expression analysis. Here there are a number of genes were upregulated and downregulated. So there are about 7,142 genes were upregulated and 3,918 genes were downregulated. Here we screened the all the genes for known function. Among this, 2,334 genes were of known gene function, and those were upregulated, and 1,524 genes were downregulated. So again, we screened these genes for the specific to pathogen response. Here we got 367 genes were upregulated due to pathogen response, and 106 were downregulated due to pathogen response.
You have to wind up. Your time is up.
Okay. So among these gene ontology revealed that all the upregulated genes were confined to cellular components. These are the number of genes which are upregulated. Interestingly, the movement core protein was the sole reason, which was 24 folds increase in the host. So these are the list of downregulated genes where it is almost confined to the cellular portions, that is chlorophyll A and A binding protein and 15S ribosomal protein. So with this, I conclude the work. The diagnosis may important in any detection of the plant diseases. So purification of recombinant protein helps in increasing specificity for production of molecular antibody. These diagnostic tools helpful in early detection, and transcriptome analysis so provides basic studying molecular mechanism associated with host pathogen interaction, and also to design the resistance varieties. So lastly, I want to thank my mentors and ISMPP organizers for giving me opportunity to present my work. Thank you, one and all.
Thank you, Dr. Priya Naganur, for your nice presentation regarding the new technologies identification. I hope judges note down the time.
Sir, she has taken 1.5 minutes extra, sir. Judges, please note down.
In this women presentation, women scientist presentation, they are taking 1.5 to 2.5 minutes extra. I don't know. Let it be last presenter. I think first time we called Durga Devi, she was not there. I hope she is now. She's.
So, yes, sir, I'm here.
Okay, madam. Can you go ahead with your presentations?
Thank you, sir.
Please. Go ahead.
Thank you, sir. Is it visible and audible? Organizing Secretary, Chairman and Coach.
Please, from the Secretariat.
Share your screen, madam.
Share your slides, madam. Please, Durga Devi, madam. Dr. Durga Devi.
Yes, sir.
You share your slides first.
Sir, is it visible?
Yes, madam. It is visible.
Okay, sir. Thank you, sir. Good morning, one and all. Myself, Dr. Durga Devi. I am working as a women scientist in the DST Sanction project. Today, my research topic on unravel the potential of endobiotic bacterium mediated resistance against Rhizoctonia solani, the instead of sheath blight disease in rice. We all know that India is the second largest producer in the rice cultivation, which accounts 117.47 million metric tons, which accounts 22.81% in the global production. Diseases are the major constraints. We all know that the sheath blight, which is next to the blast disease, it could cause the yield loss up to 50%. Still now, no resistant cultivars was developed. The Rhizoctonia solani, which has wide host range, it would be able to attack the cereals, cotton, vegetables, and pulses, etc. Available fungicides are
continuous use of chemical fungicides which leads to health hazards. The Rhizoctonia solani concern, the pathogen probably exists in the soil for a longer time, and it could taken up for the next season crop also. So continuous use of chemical causes residual toxicity, and repeated usages will be the cost effective also. So in order to search for the alternative, the biocontrol management is the possible way. So the following activities was laid out with the survey and isolation of endophytic bacteria and screening against the Rhizoctonia solani pathogen. In this, we have survey was undertaken in the different districts of Tamil Nadu, and in the different districts, Tirunelveli district, Ambasamudram, which recorded the highest disease incidence up to 69.93%. We have isolated 25 isolates. Among 25 isolates, we have isolated using tissue segment method. In this picture, we could see the pure culture of Rhizoctonia solani, and further we confirmed through the morphological, biochemical and molecular characterization. As a pathologist, we all know that it's a separate mycelium with a multinucleate condition, and it could able to form, it's also form a sclerotia. And further we proved the pathogenicity this is the symptom in the field condition, greenish gray lesions on the panicle and also on the sheath. We could see the sclerotia on the sheath also. These are the symptoms observed on the field, and again we confirmed the pathogenicity in the part culture condition. We used a BBT 5204 cultivar, and different inoculation methods are available. We used a standard mycelial plug method. Actively growing 5 days old culture of Rhizoctonia solani was inoculated in the column region of stem, and after 5 days of inoculation, we have seen the symptoms. The RS12 recorded the highest disease relative lesion height, which accounts 75.96% of relative lesion height. This is the virulent isolate, and in this picture we could see the morphological differentiation of Rhizoctonia solani. Further, in order to confirm as a molecular level, we have amplified the universal region, and we used the ITS region, and all the isolates could be able to amplify at 650 region and confirm. And further, in order to identify at the species level, we used a genus-specific primer AG11A, and it also amplified at 265 base pair. Further, we have submitted in NCB sequences, and the sequence similarity was obtained. And to isolate the endophytic bacteria from the rhizosphere and stem and different rice tissues, we have isolated totally 80 endophytic bacillus were isolated from the culm, stem and leaf regions of rice. And most among the 80, the 20 endophytic bacillus isolates which could incubate the pathogen, more than 46% incubation was observed. And in this table, we could see the antagonistic effect of endophytic against the Rhizoctonia solani mycelial growth. The EBPBS4, which recorded the highest inhibition percentage, 58.89%, which is significant, which was on par with the EBAM1 isolate, which also recorded 57.77% inhibition with the inhibition zone of 22 mm. Compared to control. In this slide, we could see the antagonistic effect of endophytes against Rhizoctonia solani. In this picture, we could see the EBPBS4 isolate. It could able to degrade the mycelial growth of the Rhizoctonia solani, and followed by EBAM1. And in order to analyze the plant growth promoting triads of the endophytic bacteria, principal compound analysis has been carried out. And in this picture, we could see the amount of variation of 20 endophytes with their PGP triads, which account 78.19% variation. These are the triads. We have screened gibberellic acid, cytoguanine, protease, beta-1,3 glucanase, hydrogen cyanide, citrophore, IgA, and salicylic acid. And in the right corner of the principal compound, that grouped the endophytes which possess the high gibberellic acid and the PGP triads, that is EBPBS4 isolate. We showed on the right corner of the principal compound analysis graph, and and further the growth promotion activity was
carried out in the roll towel method, and which recorded the 100% germination percentage that isolate and increased root length, shoot length, and vigor index compared to that control, followed by the EBAM1 isolate. And these are the pictorial presentation of that roll towel method, and we have carried out the scanning electron microscopic view to prove the growth promotion ability of the endophytic isolate. Here we could see the early initiation of the plumule in the seeds treated with the bacterial suspension of EBPBS4, which was initiated earlier than the untreated control. And the PGP traits I already explained in the PCA analysis that isolate which recorded highest cedrophore and HCN, significant producer of HCN and cedrophore and salicylic acid, which was estimated qualitative and quantitatively. And here in this table, the presence and absence was recorded with the quantitative data, which recorded 6.2% of 6.2 micromolar of cedrophore and 16 nanomole of HCN, followed by 10.47 of salicylic acid. Further, to confirm that isolate as a bacillus, universal ITS primer was used to amplify the region. The PCR amplification was performed. It could able to amplify at 546 base pair, and all the isolates amplify the region. Further, the sequences were done, and with the NCB sequence inverting was done with the NCB databases, and the potential 5 isolate based on the in vitro and antagonistic activity, growth promotion activity, we have selected screened the 5 isolate and sequenced, and the sequence similarity varied from 99 to 100% with the NCB databases, and we identified as bacillus subtilis, cirrus, and amino liquefacients. This is the phylogenetic tree construction using the mega software, and here the bacillus subtilis EBPBS4 was similar with the 100% similarity with the bacillus amino liquefacients and the bacillus subtilis strains. Again, we have screened that bacterial isolates for the antibiotic and useful biosynthetic genes. The crude antibiotic were extracted from that bacteria, and we have screened that crude in the agar-well diffusion assay. It inhibits the mycelial growth up to 40.5% compared to that of control, which accounts for an inhibition of 55%. In this picture, we could see the inhibitory effect of all the bacterial isolates, that is the potential isolates compared with that of control. It degrades the mycelial growth of the pathogen, and followed by the crude metabolites was placed on the TLC plate with the standard iturin and surfactant, and it recorded the RF value of 0.7 and 0.3, which presents the compound of iturin and surfactant. Further, the antifungal protein has been extracted from that crude metabolite using amino sulfate fractionation. The different amino sulfate fractionation with 30, 40, 50, 80, and 90% fractionation, and medium range molecular marker was used. The antifungal protein we could observe at 14 kDa and 43 kDa and 68 kDa. The 43 kDa it's present in all the 1, 2, and 4 fractionation. This is the basisubin compound which is present in the bacterial isolate. And further, in order to identify the antibiotic biosynthetic gene in the endophytic bacillus, the 20 promising isolate which recorded the highest inhibition in the in vitro, we have taken the DNA and extracted and used the antibiotic specific primers for the amplification of antibiotic genes. We used iturin, surfactant, bacillicin, fengicin, and mycosupplement gene, and most of the EBPBS4 isolate could amplify most of the antibiotics which is on par with the EBAM1 isolate. So this is the pictorial representation of all the antifungal protein amplification in the bacterial isolates. Various range of amplicons was observed in that PCR reactions with that respective biosynthetic genes, iturin and surfactant. Iturin different groups are there, A, C, D, and D, and most of the isolates could be able to amplify the antibiotic genes. These are the pictures.
And further, in order to quantitatively estimate the uterine and surfactant level, the protein was characterized in the high performance liquid chromatography. The uterine compound in the sample, that is the EBPBS4 potent isolate, is 13.62 microgram per microliter, and surfactant was 540.17 microgram per microliter, which recorded the presence of uterine and surfactant. These two antibiotics are the responsible for the antagonistic activity of plant pathogenic fungi. And further, the microtic enzymes, we have screened for the microtic enzyme presence, and most of the isolates could be able to amplify the chitinase and glucanase genes. These genes, if these genes are present, it could able to degrade the fungal cell wall of fungal cell wall. So in this table, we could see the EPPBS4 recorded the chitinase of micromole of per minute per gram of protein, and glucanase it recorded 18.93%, which is significant over the control. And the respective genes, we have specific primer used for the amplification of chitinase and glucanase gene, and mostly on the 20 isolates we have used promising isolates. It able to amplify most of the isolates. In among the 19 isolates amplified the glucanase region, and 12 isolates has amplified the chitinase gene by city clusters. And AA gene, AA is a quorum punching gene, which is which is used in the synthesis of which is regarding the quorum sensing signal molecule, that is acyl homoserine lactanase is a quorum sensing lignase molecule. The AA gene is responsible for the degradation of AHL molecule, which is present in the pathogen. So the presence of AA gene is the added advantage. So in order to identify the presence of AA gene in the potential microbes, we have used the specific primer to amplify the AA region, and most of the isolates amplified at the 800 base pair, and it could confirm the presence of AA gene in the promising isolates. And the quorum sensing activity, we have used two strains, Agrobacterium tumefaciens A136 and NT1. These are the indicator strain which is used for the sensing of quorum sensing activity, which was streaked on par with the EBPBS4 isolate, and which showed positive for that indication of blue color, which showed the positive for the presence of quorum sensing molecule in the endophytic bacteria. The next one is the formulation. This is the farmers in need of our the formulation. Already available equivalent torque formulation has a short shelf life, and farmers are not sure that the product quality and the improved formulation and delivery systems are the need of the hour. So we have developed the endospore based formulation. We standardize the medium for the sporulation of bacillus isolate, that is bacillus subtilis EBPBS4 strain, with the additives, sweeteners, anti-kicking agent, and alanine germinant receptors. We have added calcium dipiclonic acid in the medium, and we have given the stress for the production of endospore, and we formulated as a powder, and we confirmed that the presence of a bacillus through the scanning electron microscope. In this picture, we could see the presence of bacterial cell in the scanning electron microscope. And the endospore formulation in the sporulation medium, after 48 hours, 48 to 72 hours, most of the vegetary cells are converted into endospores, and after 120 hours, it achieved 13.74 log 10 CFU per ml endospore, which was it was further tested with the other broths, nutrient broth with minerals and glucose, and standard sporulation medium, which is commercially available standard sporulation medium. So in this slide, we concluded the sporulation medium was able to produce more number of endospores in 120 hours. In this graph, we could see the number of 13.74 endospore colonies are formed per ml of spore suspension on 120 hours after inoculation. So we have confirmed the endospore presence in the phase contrast microscope. We used the vanillite green, and we stained with the saffronin. After 12 hours of the endospores are present, we could see the stained bacteria in the formulation. And
your time over. Please wind up.
okay, sir. unravel the molecular mechanism of disease resistance. This is the main objective of our study. And what we the EBPS4 strain was applied, the formulation was applied in the plant as a sequential application, seed treatment, sealing dip, followed by foliar application. And we 15 differentially expressed protein was identified, and most of the proteins are defense protein. And we concluded that the promising isolate could able with the antagonistic activity, growth promotion, and it also contains the most of the defense genes which could be formulated. And we have tested the bioefficacy in the field condition, and it also recorded the maximum percent disease reduction over control, so 89% compared to the tough chemical and untreated control, and which also recorded the highest yield. So once again, I thank you, sir, for this opportunity. This is the schematic representation of our EBPBS4 model, which activated most of the different genes in the plant system, and which is the potent biocontrol agents against the management of rice sheath blight pathogens, sir. Thank you, and I would like to acknowledge the chairman Ravichandra sir, and the PA Hari sir, and Department of Plant Pathology, teaching seniors and juniors, teaching staffs, non-teaching members, and the organizing secretary to giving a nice opportunity to present my topic, sir. Thank you, sir.
For the information of judges, she has taken 2 minutes extra. Please note down.
Okay, thank you, Dr. Durga Devi Madam, for your nice presentation. I think by this we have end of the all the 5 participants who have listed for the Dr. Guman Devi Verma Women Scientist Award. All the 5 starting from Sneha, Sangeeta, Shweta, Priya, and Durga Devi, all of them have presented nicely, and they have covered the topic exhaustively. I think except one, all the women scientists have taken 2 minutes, 1 to 2.5 minutes extra. It looks to be, I think, organizing secretaries from next onward, they have to think of that one. Anyway, I especially thanks to the organizers, especially to Dr. R.K. Mehta, organizing secretary, and his team for giving me an opportunity to chair this, both the PP Singhal Memorial PI Industries Award and Dr. Guman Devi Verma Award for, I think, all the 5 presenters, earlier 3,
Sir,
and yes, ma'am.
Sir, for co-chairman, you can give the opportunity
yes.
for his remarks.
I think I thank Dr. Mehta and his team, and I request Dr. Co-Chairman Kantraj to offer his comments. Before that, I thank both the rapporteurs and co-chair. Please, Dr. Kantraj.
Yes, sir. Thank you, Dr. Egbote, sir. So first of all, once again, I thank the organizer. Yes, sir, are you hearing?
Yes, hearing.
Yes. I once again congratulate the organizers, especially Dr. R.K. Mehta sir and his team, so for organizing very excellent session for both the awards. And I also congratulate and all the best for all the participants, though actively participated. So for first award, that is PP Single Memorial Award. So there were 3 participants, Dr. Praveen, Mainuddin, and Gurudat Tegde. All 3 have very excellently, they have presented and completed within the time schedule they have given. And there was a very active participation as seen from different parts of the country, from Kashmir to Karnataka, it seems. And there was a very tough competition also, and covering very nice topics and of the research they have done. So I once again wish all the best for the participants. And for second award, Srimati Guman Devi Verma Memorial Best Woman Scientist Award, there were 5 participants. And I think all of them have excellently presented, and time was very short for them because of lot of work they have been done in different parts or different different research work they have done in different crops. And this is a very tough competition for the judges to select also, because all of them done a very excellent work. So I once again I say wish all the best for 5 scientists they have presented the work. So thank you, sir, once again all the best. So over to Dr. Egbote, sir.
Thank you, Dr. Kantraj. I think
yes,
from the central secretariat from the Bagalkot, they have to speak. I think we have completed within time
Yeah.
allotted within 12 p.m. So except one, all of you have presented. I think over to the secretariat.
Now, on the behalf of organizing committee, I would like to thank the chairman and co-chairman for leading the session and also the rapporteurs. I would also like to thank and congratulate all the participants for their enthusiastic participation. Now, the second session of the day, that is ISMPP award competitions, which includes PR Verma Awards competitions for MSc and PhD. This session is being chaired by Dr. Mahesh Tatke, Professor of Plant Pathology, Vasantrao Naik Marthwada, Krishi Vidyapith, Parbani, and co-chaired by Dr. Manoranjitam SK, Associate Professor of Plant Pathology, TNU, Coimbatore. The rapporteurs for the session are Dr. Arun Kumar KT, Assistant Professor of Plant Pathology, College of Horticulture, Bidar, and Dr. Manjunath Reddy TD, Assistant Professor of Plant Pathology, College of Horticulture, Kolar. The chairperson of the session, Dr. Mahesh Tatke, has brought about immense contributions to the field of plant pathology and has published 16 research papers, 35 technical papers, and many popular articles. His significant contributions include ad hoc recommendations for the management of soybean rust and Sigatoka disease of banana. And the co-chairperson of the session, Dr. S.K. Manoranjitam, has 14 years of research experience in plant virology. She is credited with the release of 4 varieties and 6 technologies from TNAU. She is the recipient of 12 awards and has published 26 research articles in journals of national and international repute. Now, I request the chairperson, Dr. Mahesh Tatke, to take over the session. Sir, please.
Thank you, madam. Thank you, one and all. So, I will welcome my co-chairman, Dr. Manoranjitam SK, to join.
Sir, good morning.
Good morning. Then I will also invite Dr. Arun Kumar KT and Dr. Manjunath Reddy to join with me for this session.
Sir, one submission, sir. There is slight change in the report here. Arun Kumar is not there. Dr. Prashant from College of Horticulture, Arabavi. He will be the rapporteur.
Okay, Dr. Prashant, I will request you to please join for this session along with me as a rapporteur. Okay, with the permission of chair, I would like to start this session. Shall I start, sir, with the permission of you? Yeah, okay. So for this PR Verma Award competition for MSc student, there are total 7 participants are participating in this contest. Before announcing their names, I have some instructions for those competitors. The first instruction is your time allotted for this one is 10 minutes for your presentation. You will get 10 minutes time. After 8 minutes, there will be one bell for indication that your last 2 minutes are remaining. If you take extra time, that will be negative impact on your presentation. So please keep this time schedule in your mind and accordingly try to finish up within the time. So now I will read the names of participants, those who are contesting for this competition. So first is Girisha D. After that R. Ovia. Then the third number S. Mukhopadhyay. Then the fourth student is Sindhu Shri DR. Fifth is Sri Narayana B. Sixth is Gutha Venkat Ramesh. And the 7th student is Nagesh. So by this sequence only our presentations will go on. So I will first invite.
Sir, one more submission. Sir, Chairman, sir, all the participants should put on their video. They should put on the
all the participants, they will put on their video so that they should be on the record.
Sir, Chairman, sir, kindly give a call to all the names of the participants so that just
they are so
there.
Once again I will call the name of students. Girisha D.
Yes, sir, I'm
is
present.
Present? Girisha?
Yes, sir.
Yeah, okay.
Yes, sir.
Then the next.
I'm ready, sir.
Yeah. So next is R. Ovia.
Yes, sir, I am present, sir.
Okay. Then third, yes, Mukhopadhyay. Mukhopadhyay.
Yes, sir. Present, sir. Present.
Present. Okay. Then Sindhushri DR.
Sindhushri.
Sindhushri DR.
Okay, may not be there right now. Let her inform her when she joins.
Then the Srinarayan B.
Srinarayan, I'm present.
Yeah.
So
Then next is.
I'm unable to put on my video.
Yeah, put on your video, please.
Sir, it is.
No, not now while presenting.
Oh, okay, sir.
Then Gutha Venkat Ramesh.
Sir, I am there, sir, and ready to present, sir.
Okay. Then Nagesh.
Yes, sir. Present, sir.
Okay. So once again, I will repeat the instructions for all of you. The time allotted for your presentation is 10 minutes. After 8 minutes, one short bell will ring. You have to finish your presentation within 10 minutes. If you take extra time, that will be the negative impact on your presentation. Okay. So shall we start now?
Yes,
so
sir.
I will call on first Girisha D to present his research topic for the award competition. And before starting him, I wish all the competitors best luck for them. Okay. So Girisha, you can start now.
Yes, sir. Shall I start, sir?
Yeah. Your time starts now.
Okay. So my screen is visible, sir?
Yes, you can share your screen.
Sir,
share your
already
screen.
shared.
Okay. No, it's not visible. You unshare and again share it.
Okay, sir.
It's not visible.
So now it's visible now, sir?
Girish has started. Yeah, now it is visible. So you can start it now.
Okay, sir. Very good morning to all of you. Myself Girish, MSc scholar from Department of Plant Pathology, US Darwin. I work under the guidance of Dr. P.V. Patil. It's my privilege to present my MSc research work on loss assessment and integrated management of root rot of sugar beet caused by Sclerotium ralsei for the prestigious PR Verma Award. Let's coming to introduction. Sugar beet, it's belong to family Chenopodiaceae. It's originated in Mediterranean region. Sugar beets are produced approximately in 8.5 million hectare area in 60 countries around the world. Currently, sugar beet account for 25% of world supply of sugar. In India, especially in northern India, temperate sugar beets are being cultivated in 1980s, but recently tropical sugar beets have been introduced into southern parts of India, and their cultivation has been carried out from 8 to 10 years. Tropical sugar beet is now emerged as a commercial field crop because of the favorable characters like a shorter duration of 5 to 6 months, and it needs a moderate water requirement of 60 to 80 centimeter, high sugar content of 15 to 17%, and sugar recovery of 12 to 14%. And it also improves the soil condition because of the tuber crop. It grows well in saline and alkali soils and helps in continuous functioning of the sugar mills. Here we can see the top 10 sugar beet producing countries in the world. Russia stands first with a production of 39.2 million tons, followed by France and USA. Sugar beet is susceptible to vast number of the disease caused by fungi, bacteria, nematode, and viruses. Among them, one of the important soil-borne disease, root rot of sugar beet caused by Sclerotium ralsei, it will cause a very much damage to the roots. And this disease is a limiting factors in the cultivation of sugar beet in southern USA, in warmer and humid areas of Europe, in India, and some parts of Asia. In India, this disease was first reported in 1965 by Singh at Pantnagar, Uttarakhand. It causes 14 to 59% losses in root yield and reduces sugar content up to 20% in certain varieties under favorable conditions. Because of the omnivorous and facultative nature of the pathogen, it is whether difficult to control the disease caused by pathogen. While the fungicides are the most common tools for the controlling the disease, in recent years there is a growing concern over their usage because of their potential hazardous effect on soil biota, particularly on soil antagonists. These factors led to a search for the innovative management practices for management of this disease. Information regarding management of sclerotian ralsei in tropical sugar beet in India using biocontrol agent and plant-based products is very meager, as the present investigation was taken up with the following objectives. Here you can see the objectives of investigation: isolation of biocontrol agents from rhizosphere soil of sugar beet from different parts of India, and in vitro evaluation of isolated biocontrol agents and alkalic extract against sclerotian ralsei, integrated management of the disease, and to study the reaction of sugar beet varieties to root rot and estimation of sugar yield losses. The pathogen was isolated from infected plant. The pathogen produces a white tuft radiating mycelium. It also produces a brownish sclerotial bodies, and it's also produced a septate and branched mycelium. By all these cultural morphological characters, the pathogen was confirmed as sclerotian ralsei. Here we can see the field symptoms of root rot of sugar beet caused by sclerotian ralsei: initial wilting and drying of the plant, and complete wilting and drying. Here you can see the mycelial growth at the collar region and formation of whitish mycelial growth on root surface. Finally, formation of sclerotial bodies, and the dead and decayed plant are susceptible to secondary infestation. Let's come to first objective: isolation of biocontrol agents. For isolation of different biocontrol agent, 18 rhizosphere soil samples from different sugar beet growing areas of India were collected and used for isolation of biocontrol agents. 10 soil samples were collected from Amritsar, Punjab, 6 soil samples were collected from Baramati, Pune, Maharashtra, and 1 soil sample collected from Indian Institute of Sugarcane Research Institute, Lucknow, UP, and 1 from MARS, US, Dharwad. A total of 6 trichodoma isolates and 1 trichodoma viride isolate and 16 Bacillus subtilis isolates were isolated and identified based on cultural and morphological character. Here you can see the cultural and morphological characters of trichodoma arzianum isolates. Colonies were growing rapidly, bright to dull green, yellowish green, and white green. There is a variation in color among the isolates. And the mycelium was septate, align, branched, and conidiophore shows a frequent branching, verticillate, and filaments are short, narrow at the base and pointed at the neck on verticillate. They are up to 3 to 5 number. Conidia are subglobous to ovoid in shape, produced at the tip of the fillet and dark green color. And conidiophores also produced infrequent and producing both in terminally and intercalary. Here you can see the cultural and morphological characters of trichodoma viride isolate. Colonies are fast growing, light green to dark green in color. Conidiophores with infrequent branching produced in compact, and main branch produce several side branches. All the branches stand at right angle. Fillets arise in false orals. There are 2 to 3 in number, narrow at the base, widening at the middle, and abruptly pointed at the neck. This picture represents the pure cultures of trichodoma isolates. Here you can see the phyllets and phylophore structure of different trichodoma isolates. For confirmation of bacterial biogen, Bacillus subtilis, a different morphological character was studied. Those are colony colors, cell shape, Gram reaction, and biochemical characters like starch hydrolysis, VP test, citrate test, gelatin liquefaction, casein hydrolysis, and by the biogens are studied, and results are presented here. Here you can see the different morphological characters. First we can see the colony color on nutrient agar medium. The colony color range from cream white to dull white and yellowish white, and all the colonies produce green color and ichrome bacillus medium. The colony shape varies from circular to irregular, and all the isolates produce rod shaped cells. They are showed positive response for Gram reaction and presence of endospores. These are the pure cultures of Bacillus subtilis isolates and nutrient agar medium. This picture represents the Gram positive reaction on rod shaped cells of Bacillus subtilis isolates and endospore staining. Let's come into biochemical and functional characterization. All the isolates showed positive response for various biochemical tests conducted for starch hydrolysis, VP test, citrate test, gelatin liquefaction, casein hydrolysis, catalase test, and phosphate solubilization. These are the pictorial representation which represent a positive response for various biochemical tests conducted. Let's come to second objective: in vitro evaluation of isolated biocontrol agent and alkalic extract against sclerotian ralsei. Among the different trichodoma isolates evaluated under in vitro condition against the growth of sclerotian ralsei by dual culture technique, the isolate trichodoma arzianum from Institute of Organic Farming USD isolate, which is recorded 69.41% of inhibition of mycelial growth, and the least inhibition was recorded in trichodoma viride Amritsar isolate 5. This picture represents the inhibition of mycelial growth by trichodoma viride and arzianum isolates. For different 17 Bacillus subtilis isolates isolated from rhizosphere soil sample sugar beet, tested under in vitro condition. Among them, Bacillus subtilis ambrosial isolate 6 has recorded 48.92% inhibition of mycelial growth, and least inhibition was recorded in Bacillus subtilis ambrosial isolate 8, which is of 8.06%. This picture represents the inhibition of mycelial growth by Bacillus subtilis isolates. Let's coming to in vitro evaluation of various oil cake extract against Sclerotium ralsei. Different 6 oil cake extracts were tested against the mycelial growth of Sclerotium ralsei by using Poisson food technique at 5, 10, and 15% concentration. Irrespective of the concentration tested, the Pongame cake 2, which is collected from a biofuel unit US Dharwad, has recorded 40.39% inhibition of mycelial growth, followed by Pongame cake 1 and neem cake. And there is no inhibition of mycelial growth that was observed in groundnut cake and Simaruba oil cake extracts. This picture represents the inhibition of mycelial growth of Sclerotium ralsei by oil cake extracts. Let's come to integrated management of the disease. A field experiment was conducted during Rabi 2019-20 at Main Agriculture Research Station, University of Agriculture Science, Dharwad, by using 11 different treatment and 3 replication, and the variety used was SP001. Here you can see the different treatment details. The treatments are designed by using already recommended chemical tebuconazole 250 EC and in vitro effective alkanes and which are Pongamia and Nema alkane, and in vitro effective biogens which are Bacillus subtilis ambrosial isolate 6 and Trichoderma arzeanum IOF isolate. The observation on seedling emergence at 15 days after sowing was recorded. Here you can see there is no significant difference among the different treatments with respect to seedling emergence. But the treatment T1 involving basal application of Pongamia cake enriched with Trichoderma arzeanum IOF isolate has recorded highest seedling emergence of 76.22%, and the disease incidence at 30, 60, 90, and at the time of harvest was recorded and calculated by using total number of plants and number of plants infected. At 30 days after sowing, least root rot incidence was recorded in treatment 1 and treatment 9, and highest incidence was recorded in untreated control. At 60 days after sowing also, a least root rot incidence of 0.69% was recorded in treatment T9 involving seed treatment plus drenching with tebuconazole 250 EC at 30 and 60 days after sowing. Let's see the mean total percent disease incidence. In a least root rot incidence of 12.96% was observed in treatment T9 involving seed treatment with tebuconazole 2% DS and followed by drenching with tebuconazole at 30 and 60
Your time is up. Please wind up. Girish.
Okay, sir.
Conclude.
To study the reaction of sugar beet varieties, all the 3 sugar beet varieties indicated susceptibility to root rot. For estimation of sugar yield losses, the different sugar beet varieties were categorized based on different degree of rotting. And at 100% rotting, all the 3 sugar beet varieties has recorded least Brix percent. It is estimated by using hand refractometer. Let's coming to summary and conclusion. I want to conclude by saying the integration of fungicide along with biogens and plant-based extracts was very useful in controlling the disease. Thank you, sir. I'm thankful to ISMPP and UHS Bagalkot for this opportunity.
He has taken 1 minute extra. Judges, please note down.
Yeah. So, thank you. And here I would like to announce one small instruction. As this is online session, there will be no questions and no discussion on the topic presented by the contestants. Thank you. Now I will call on second candidate, that is R. Ovia, to present the topic for this PR Verma Award.
Yes, sir.
Your time starts now.
Sir, is this visible, sir?
Yeah, visible. Yes,
A
starting.
pleasant afternoon to all. Myself, R. Ovia from Tamil Nadu Agriculture University. I'm going to present on the topic glyotoxin producing Trichodoma virens effectively suppress the dry root rot of black gram. As we all know, this Trichodoma species of beneficial soil saprophytes, which are commonly used for the management of the soil-borne plant pathogens. These suppress the phytopathogens through various mechanisms like mycoparasitism, antibiosis, lysis, etc. And these Trichodoma species produce several antibiotics, among which the glyotoxin was a well-known antibiotic, and it is also identified as a secondary antibiotic next to that of the penicillin. In earlier days, this glyotoxin and the glyotoxin-producing Trichodoma has been commercialized, and it is is available in a name of glyogod and soil god. So far, there is no study on its behavior and persistence in the soil ecosystem for assessing its biosafety and bioefficacy. That too, there is no study on assessing its biosafety and bioefficacy on the soil-borne pathogen, and hence our study is focused on the objectives of this: isolation of glyotoxin producing Trichodoma virens strain, and analysis of glyotoxin stability behavior in different culture medias, irrigation water and soil ecosystem. And the main objective is to test its bioefficacy against the soil-borne pathogens. First, I have been isolated up to 37 trichodoma isolates. These are different species of trichodoma isolates, and this is isolated from the trichodoma selective medium. And these isolates are subjected to the TLC analysis in order to verify whether these produce glyotoxin or not. But here, all the trichodoma isolates which I have been isolated first, that is trichodoma isolate 37, none of them have not produced the glyotoxin with that of the standard. Then, in order to identify and to isolate the glyotoxin producing Trichodoma virens strain and to purify the glyotoxin in a larger scale, we have purchased 2 culture from the microbial type culture collection that from Chandigarh. The 2 strains, namely 2194 and 2977, and from these strain I have been purified the glyotoxin, and using that purified glyotoxin, I have amended that purified glyotoxin in an PDA medium and isolated the glyotoxin producing Trichodoma virens strains. And here it is. These are the 10 Trichodoma virens strains which have been isolated from the glyotoxin amended PDA medium, which is shown here along with that of the reference strain 2977 and 2194. And below, bottom picture of the TLC picture, which represents the level of production of glyotoxin by various Trichodoma isolates. Among them, TK2 is an higher producer glyotoxin producing strain, which shown the higher level of the glyotoxin production with reference to that of the 2977, that is in reference strains. And we have also found another easy method for the identification of the Trichodoma virens, that is based on the conioidal morphology. The glyotoxin non-producing Trichodoma type shows a Trichodoma type of conidiophore. That is, in an highlighted area, you can note that the conidiophore, which bears the phyllates at the tip at a right angle. These are the Trichodoma morphology which doesn't produce a glyotoxin, whereas the Trichodoma virens, which produce the glyotoxin, has a conidiophore morphology of a glyocladium type. That is, at the tip of the conidiophore, a tuft of phyllates are present there, and these are arranged in an acute angle. And further, it is also confirmed using the PCR amplification, and the strain TK2 is amplified as an amplicon range of 650 base pair, and it is also confirmed by the NCBI blast search, and it shows an identity of 90 to 70%. And in order to dissipate the fate of the glyotoxin production, accumulation, and dissipation, the glyotoxin producing Trichodoma strain is inoculated in the Weinling medium for about 15 days inoculation period. And this in inoculation period, the samples were taken at different time intervals and the TLC was spotted. This experiment results that the glyotoxin production starts from the 2nd day and lasts up to the 6th day. And there was no glyotoxin production at the 1st day, and the glyotoxin production was stopped at the 8th day, and there was no presence of glyotoxin on 12th and the 15th day. And interestingly, another compound which is produced on the 8th day, this compound production starts with the production of the glyotoxin stops, and this compound keep on increasing gradually. And the left side picture of the mycelial dry weight, which represents that the logarithmic phase of the glyotoxin, that is, at the time of the glyotoxin production, the mycelial phase has been started, and when the glyotoxin production reaches the maximum, the mycelial growth rate also reaches a maximum. Hence, here it is indirectly inferred that unlike that of the other secondary metabolite, the glyotoxin has not been produced on the lateral growth stage, but it has been produced on the earlier logarithmic phase. And here it is further confirmed that the presence of the glyotoxin is confirmed using the HPLC, using the standard and the sample along the peak area. And then I have said before itself in that picture now, an interesting unknown compound has been produced at the stopping of the glyotoxin, and that compound is purified again using the LCMS analysis, and based on the Dalton range of 357, it is identified as methyl thioglyotoxin. Here what happens is that this trichoderma, which itself produces an enzyme methyl transferase and converts its production of glyotoxin into an methyl thioglyotoxin over a time period. And in order to see the effects of pH on the stability of the Weinling medium at different pH levels, various pH levels of acidic to alkaline has been inoculated, and from that it is inferred that acidic pH, that is lower the pH, higher the gliotoxin production. And you can from this picture you can note that in the alkaline pH, that is 6 and 7, there is a smear band. This smear band represents that the gliotoxin produce itself, and suddenly it converts into a degraded gliotoxin. And again, normally in a liquid bioformulation, molasses yeast medium and PDA medium was used for many organisms. But this gliotoxin is produced only in the Weinling medium. Hence, we have to check whether this gliotoxin can be commercially formalized in a liquid bioformulation or not. So we have inoculated this gliotoxin producing trichodoma strains in the molasses yeast medium and PDA medium and the Weinling medium. Upon these three mediums, in molasses yeast medium, the gliotoxin production was stable, and the stability which increases up to many days, that is 30 days, whereas in PDA and in Weinling medium, the production was less when compared to that of the Weinling medium, molasses yeast medium. And then here it is an important study that effect of pH on the irrigation water, that is culture-free irrigation water. So culture-free irrigation water is taken, and it is spiked with a considerable amount of gliotoxin at various pH level, and it is incubated for different days of inoculation, and the results were analyzed. Upon analysis, it is seen that this culture-free irrigation water, there is no degradation of gliotoxin. That is, the degradation the gliotoxin start to degrade, but the degradation occurs over the 20th day. And the stability of gliotoxin, here this picture infers that the higher soil moisture, complex microbiota. Then this picture shows the in vitro effect of the gliotoxin, that is gliotoxin at the concentration of 50 and 100 inhibit the macrophomina. And then it has no phytotoxic effect, and it is shown in this picture. And it is an in vitro growth assay in dual plate culture. And upon the in vitro assay of the culture filtrate, you can notice the red color indicates the culture filtrate of non-producing, which does not inhibit, whereas producing inhibits highly. And this is a growth promoting activity. In the table, you can refer it promotes the growth. And finally, when subjecting to that of the pot culture experiment, the gliotoxin high producing Trichodoma virens strains have been effectively controlled this dry root rot of black gram. And here is the statistical evidence for it. That is dry root of black gram is effectively controlled by the gliotoxin producing Trichodoma species. And I would like to conclude my presentation by summarizing it. Only the Trichodoma virens produce the gliotoxin, whereas others do not. And the gliotoxin is quickly degraded in alkali condition, whereas it slowly degrades and takes longer duration in the acidic condition. And it is highly stable in culture-free irrigated water, and it is stable in moderately wet and dry conditions. I would like to conclude my presentation by saying that gliotoxin and the gliotoxin producing Trichodoma virens are highly effective on the management of the soil-borne diseases, that too especially on the management of the dry root rot of black gram. And I would finally acknowledge that I would privilege to acknowledge the financial support provided by the BARC Mumbai, and I would like to thank my chairman and all the dignitaries who are for the patient listening for my presentation. Thank you one and all. Thank you.
Thank you, Ovia, for the timely presenting your presentation in time. So now I will call upon the third contestant, Yes Mukhopadhyay, to present his.
Sir, for information of judges, he has taken 9 minutes.
How much?
9 minutes, sir. 9 minutes.
Yeah, 8 plus 1. 8 plus 1.
Yes, sir. Sir, please proceed.
Sir, I will call upon the third contestant, Yes Mukhopadhyay.
Sir, I am.
Yeah, your time starts now.
Good afternoon, everybody. Myself, Sabyasachi Mukhopadhyay. Today, I will going to present my presentation on image processing system-based analysis of down immunology severity of cucurbits and its relationship with weather variables. Now, down immunology severity of cucurbits caused by Pseudomonas productivensis is a serious weather-sensitive and radial foliar disease for all over the world, and all the available pathotypes infect cucumber crops. And for this management of this pathogen, high-risk pathogen, several fungicidal sprays are required, and this leads to a development of the resistance. And if we delay 1 week after symptom development, then it can lead 50% yield losses. Then several practices are available, management practices are available, but without chemical, except chemical management practices, other management practices are not able to prevent the infections in the field when applied individually. But chemical preparation needs precautions and chances of development of the resistance. So my objective was to identify the influence of weather variables of sporangiophore and sporangial morphology of Pseudomonas cubensis, and to develop weather variable based disease prediction models for downy mildew disease severity of cucurbits, and to analyze downy mildew disease severity based on image processing approach. Now these are the some benefits of my approaches, and some reviewed literature which shows RGB sensors and spectral sensors are capable to detect different diseases in different crops. My method was my experiment was conducted in the C block from BCKB Koldani, and in this case disease incidence and PDI percentage was recorded throughout the year from April 2019 to March 2020 with every 15 days interval, and different attributes of sporangiophore, sporangium and other parameters are recorded, and various models are used for prediction of the disease severity in case of downy mildew disease severity of cucurbits. And in case of RGB image processing system, white background real-time captured images are used, and they are segmented out, and their disease pixels are calculated, and disease severity percentage are calculated. And these same images are used for manual disease coding by different plant pathologists with 1 to 10 disease scales. And reflectance index of 400 nanometer to 1000 nanometer are used for NDVI, PRI, and ARI vegetative index calculations, and along with this, different pigment concentration, total carbohydrate concentration, and histopathological changes are also considered. Now, result and discussion part. In case of month-wise variation of the morphology of the Pseudomonas cubensis, I opted that in the month of May 2019, it is lowest, that is sporangium length and height at which branching started, and number of branches, it is lowest, and it trends to its peak in the month of January 2020, and in the month of August, the sporangium length is highest. Some images are there. Correlation between different weather parameters shows that in case of sporangiophore length and height at which branching started, temperature range, relative humidity morning, relative humidity evening, and relative humidity average are positively correlated, whereas others are negatively correlated. In case of number of branches, only temperature range and relative humidity morning are positively correlated, and others are negatively correlated. In case of sporangium length, only the temperature range is positively correlated. Whereas based on correlation coefficients and based R-square value, different MRM models for prediction of the different morphological structures of Pseudomonas cubensis are evaluated, and they are evaluated throughout the year from April 2019 to March 2020. And for the height of the sporangiophore, Tmax and day interaction between day temperature and relative humidity maximum is important critical factor. And here R-square value is 0.883 and RMSC value is 6.76. Whereas in the height at which branching started, here RMSC value is 4.11, and in case of number of branches of sporangiophore started, it is RMSC value is 0.24. In the case of disease severity estimations, the disease severity estimated based on this multiple regression analysis model shows that different weather parameters are positively correlated except temperature range, which is negatively correlated, and the relative humidity morning and relative rainfall transformation factors are negative, non-significant at the 5% level of significance. Now, different models are compared for disease severity estimation purpose, and their R-square value and accuracy percentage are checked. In case of R and R-square value, it is highest in case of KNN model, and it is lowest in the case of RF model, that is random forest. In case of accuracy percentage, it is reverse, that is it is highest in case of random forest, and whereas followed by KNN. These are some examples of how these real-time captured images are segmented out, and the percentage of infected area are calculated, and based on this 1 to 10 rating scales, the scores provided. Now, in this case, the 77 images are used for enumeration of their disease severity by different plant pathologists and image processing based systems. Here 77 images are appeared to the in front of the different plant pathologists of different expertise level, and they enumerated their ratings, and based on this 68.80% average ratings are matching with image processing systems, whereas modal value is high in case of 90.90%. So in this from this slide we can conclude that in case of where experts are not available for these severity estimations, we can use these image processing based systems for these severity estimation purpose. Some anatomical changes are also seen in case of healthy light green, light yellow, deep yellow stage, yellow necrotic and necrotic stage. Spongy mesophyll and palisade mesophils are degrading, and number of chloroplasts are degrading with advancements of the time. In case of chlorophyll concentration, it is highest in case of healthy and degrade and it deteriorating with advancement of the time. And here it is noticeable that carotenoids concentration, that is carotene and xanthophyll, it increases because plant grade stage, and it increases up to the dark yellow stage, and followed it decreases. But in the case of carbohydrate concentrations, it is highest in the case of light green, and then it gets its another peak in the dark yellow stage, and then onwards it's decreasing. Some images are there. In case of hyperspectral imaging, I divide the total system in two groups: pre-symptom development stage and after symptom development stage. In pre-symptom development stage, in case of healthy, blue line shows the healthy healthy spot reflectance, and it is quite higher in near infrared region, that is 700 to 1000 nanometer. And it is because of healthy spongy parenchyma, spongy mesophyll cells are present in, and with advancement of that time, these spongy mesophyll cells are deteriorating. And different vegetative indices, NDVI, ERI, and PRI shows the following trends. Some images are there. In case of conclusions, maximum temporal length of the sporangiophore obtained in the month of January and February, and increases from secondly increases from May to September due to the lowering of the day and night temperature and the increase of the higher relative humidity. Height of the branching also started highest in the month of November to February, and the lowest number of branching and the lowest disease severity is obtained in the month of April to May due to the higher temperature and dry spell. Height at which branching started is it is noticeable that when disease severity is lowest, then the height at which branching started ratio is higher. Then when disease severity is more, the ratio is low. Stage-wise regression by considering all the weather variables as predictors shows that temperature and humidity get roles on the morphological parameters. These two models can be linked with fungicide scheduling in the field, and these are some critical predictors, and MRA model percentage for different morphological analysis. And these trends I already explained, and RGB based images are also already can be utilized in the field for where different experts are not available. In case of hyperspectral image signature, it is capable to detect plant infected disease plants early before the symptom production stage, and different vegetative indices are also capable to detect this. So thank you. Myself.
Okay. Thank you, Mukhopadhyay. Now I will call on the fourth contestant for PR Verma Award, Sindhusri DR.
Yes, sir. Yes, sir.
So put on your video because and share your slide share screen. You have to share.
Is it visible, sir?
Your screen is not visible. You share your screen so that we can start your time. It's not visible to us.
Yes, sir. Yes, sir.
Yes. Yeah.
So now
it is. Yeah, it is start visible now, and your
Yes.
time starts now.
Good afternoon, everyone. I'm Sindhushree, worked under the guidance of Dr. Gurudat Hegde. I feel privileged to participate in the PR Verma Award competition for MSc students on the research eco-friendly management of fungal foliar diseases of tomato by using botanicals, biogens, and ITKs under protected cultivation. Coming to its introduction, tomato is a most important commercially vegetable crop grown throughout the tropical and subtropical regions. The present status, tomato stands second under the production next to China, and in India, it's growing in an area of 0.8 million hectares with a production of 19 million tons. The tomato production is hampered by various part of playing an important role, causing a loss up to 35 to 78%. Protected cultivation nowadays farmers are going towards cultivation of crops under protected conditions. It's providing earliness and higher yield, and for organic management, considering the ill effects of the chemicals, botanicals, biogens, and indigenous technical knowledge playing an important role in management of the fungal foliar diseases. There is no much work has been done with respect to the management of fungal foliar diseases and eco-friendly and protected cultivation. Hence, the present work has been taken. The work was conducted with the following objectives: one is survey for severity of the disease and protected cultivation, and there is molecular identification, and third objective is management of fungal foliar diseases. Coming to the first objective, survey for severity of the diseases of tomato under protected cultivation. Among the 4 districts surveyed, the Haveri district found to be scored higher severity of early blight and septorial leaf spot, and higher severity of grey leaf spot and powdery mildew was found maximum in Bagalkot district. This is the symptomatology and morphological characters of alternate solani. Here you can observe the concentric ring appearance of early blight symptoms. This is the pure culture and the alternaria solani. This is the symptoms of the septorial leaves. What you can observe the circular spots and pure culture, and this is the quantity of the septorial lycopacity. This is the image of the necrotic circular lesions you can observe with the pure culture and conadia. And this is a mixed infection of the mixed infection of all the 3 diseases. This is the microscopic observations. The morphologically identified for foliar pathogens were confirmed through molecular identifications, and the Darwin isolate found 99.44% similarity with oxygen number J49, and for septorial lycopacity found 99.27% similarity with an oxygen number DQ841156, and Staphylium solani found 99% similarity with the J using botanicals, biogens, and ITKs. In vitro evaluation was conducted of botanicals against Alternaria solani and septorial lycopacity. Among the 8 botanicals used, garlic found to be the significant in inhibiting the Alternaria solani and also the septorial lycopacity. Among the in vitro evaluation of antagonistic against Alternaria solani and septorial lycopacity, among the 11 biogens used, Trichoderma azanum Darwin isolate found to be effective in both the case of Alternaria solani and also septorial lycopacity. In the in vitro spore germination by ITKs against Alternaria solani and septorial lycopacity, covering found to be effective at effective with 77.33% inhibition. Among the ITKs against septorial lycopacity evaluated, covering at the rate of 76.67% found to be the percent inhibition. Coming to the evaluation of botanicals, biogens, and ITKs against fungal foliar disease of tomato under polyhouse conditions, the experiment was conducted at the Biofarming Unit of Certified Institute of Organic Farming and University of Agricultural Sciences, Harvard. Effective botanicals, that is garlic at the rate of 15%, and biogens at the rate of biogens like Trichoderma azanum and Pseudomonas fluorescens, and ITKs covering at the rate of 15% were taken from the in vitro studies, and they were evaluated under polyhouse conditions. The first dose was given at 15 days intervals, and disease severity was recorded before and after spray. This is the experimental design. The randomized complete block design was followed with 14 treatments and 3 replications. This is an important step that everyone has to be taken for organic management. We have taken soil test-based applied soil test-based vermicompost at the rate of 2.5 tons per hectare, and neem cake at the rate of 2 tons per hectare in the ratio 10:2. Along with that, possible PSB and Trichoderma azanum, Pseudomonas fluorescens, Purpurocilum, and Metarhizium anisopliae were also added to the soil at the rate of 2 kg per hectare at 15 days before transplanting. And also we have done seed treatment and also The link dip, seed treatment with trichoderma asianum at the rate of 10 kg per 10 grams per kg of seeds, and drenching with the Pseudomonas fluorescens at the rate of 10 grams per liter. And we have also taken care of insect pest management under the polyhouse conditions. We have incorporated the sticky traps and also ferment traps for monitoring of the insect pest. The sprays of myco-insecticides like metrorrhizum, lecanicilium, pneumoria relay were given at the rate of 2 grams per kg, and also Panchagavya and the vermi wash were given through the drips at every alternate weeks. This is a table representing the results of the eco-friendly management under protected cultivations. These are the 14 treatments. There includes sole applications of the garlic, trichoderma asianum, Pseudomonas fluorescens, and also vegetable sulfur, and also sequential spraying of these. Among them, the treatment T7, garlic, Pseudomonas fluorescens, caveolin, and vegetable sulfur found to be the effective treatment, showing a 55.65% inhibition of reduction of early blight, and 51.54% reduction of septorial leaf spot, and 52.25% reduction of gray leaf spot. And this effective treatment is on par with the treatment T9. Here you can observe the effective treatment T7. This is sequential spraying of garlic, Pseudomonas fluorescens, caveolin, and vegetable sulfur. Treatment T9, and this is the untreated control. Observations were also taken on the growth and yield parameters. There was no much difference between the treatments with respect to this plant height, internodal length, number of fruit plus cluster, and number of pickings within the treatments as compared to the untreated control. It was good, and T7 is found to be the effective treatment with for all these growth parameters. Effect of different eco-friendly disease management practices on yield. The treatment T7 is found to be the very effective one with a yield of 49.40 quintals per 506, that is 5 quintals, as compared to all other treatments. And this yield is on par with the sequential spraying of Pseudomonas fluorescens, garlic, vegetable sulfur, and caveolin. And the cost economics of was also calculated for these treatments, and treatment T7 was found to be the effective. That's with 22.61 BC ratio, benefit cost ratio of about 2.61, and T9 found to be having 2.49 benefit cost ratio. These are some of the delegates who have visited my experimental plot during my research work. That is M.B. Chetty sir, N.S. Rathore sir, and M.K. Naik sir. And also some of the Dr. Shrikant Kulakarni, Dr. Brahma Singh, Dr. Raghavendra Chemist, the team of Plantics, and also Dr. M.B. Patil, Dr. Subramaniam, Dr. Yashoda Harati. I would like to thank all the delegates for their encouragement and support during my research. Finally, I would like to conclude my presentation by saying among the survey conducted, Averi found to be having maximum severity of early blight and septorial leaf spot, highest severity of septorial leaf spot, and highest severity of grey leaf spot, and the pouty mildew was found maximum in the Bagalkota district. And the morphologically identified pathogens were molecularly confirmed, and these isolates, Dartmouth isolates, were found to be having 99% similarity with the referred isolates of NCBI and sheep bank. And among the in vitro studies, garlic at the rate of 15%, and caveolin at the rate of 15%, and the trichoderma asianum found to be the effective treatment. And the T7 found to be that is sequential spraying of garlic, Pseudomonas fluorescens, caveolin, vegetable sulfur found to be the effective treatment with respect to yield and also the benefit cost ratio. Thank you. Hello.
Thank you, Sindhusri.
Thank you, sir.
So
Sir,
now I will
9 minutes.
9 minutes. So now I call on the next contestant, Sri Narayan B. to present.
Sri Nayana, sir.
Sri Narayan, sorry, Sri Nayana. Sorry for the wrong pronunciation. Sri Nayana, you can start your screen sharing so that your time.
Okay, sir. Sir, am I audible, sir?
Yeah, and your screen, yeah, screen is also visible now, and your time starts now.
Good afternoon to one and all present here. Myself, Sri Nayana from TNAU Coimbatore. Respected Chairman, Co-Chairman, rapporteur, scientist, and my dear friends, I am here to present my PR Verma Award competition entitled on Trichoderma conjuntiopsis mediated interaction, resistance, and suppression of fungal nematode complex of cucumber under protected cultivation. As we all know,
Put on your video.
sir, audio or video, sir?
Your video is not started. You are not.
Slideshow mode, please. Go put on to slideshow mode.
Your video should be started. Yeah, now it's
Sir,
visible.
shall I start from the beginning?
Now it is visible. You can continue.
Sir, shall I start from the beginning?
Pardon?
No, Naina, you proceed. Naina, you proceed. Proceed.
To proceed, your screen is also shared and your video is also on. You continue. Don't waste your time. You continue.
Sir, can I start from the beginning?
No, you will move your time. You just.
And please put the in presentation mode.
You continue from this only.
Next is at 25.50.
Sir, is it visible now?
Yes, it's visible. Please put it in presentation mode.
It is in presentation mode, ma'am.
Okay. Please proceed.
Sir, good afternoon, honorable present here. Myself, Shreena from TNU Coimbatore. I'm here to present my PR Verma Award competition entitled on Trichoderma conjuntiopsis mediated interaction, resistance, and suppression of fungal nematode complex of cucumber under protected cultivation. As we all know, cucumber is one of the most important vegetable crop which is grown worldwide. India stands fourth position in area production and productivity of cucumber apart from Russia, China, and Turkey. Cucumber is harmed by many pathogens, of which the fungal nematode complex incited by the Fusarium oxysporoformis, which is cucumerinum, and root node nematode Merodocaina incota plays a major role, which causes around 70% crop loss with 10 to 50% reduction yield. So here comes why we have gone for this topic. So when we do cucumber and fermented cultivation for several years, it results in the soil exploitation due to the indiscriminate use of fungicides and other chemicals. Later on, it leads to resistance among pathogens and results in inoculum development, which leads to the poor soil health. And when we apply biocontrol agent to this soil, which will improve the soil health by means of accelerating the microorganisms. Also, it boosts the immunity of the plants. Thereby, it results in the production and productivity of the crop. So here comes the objective one. Here we have screen.
Hello, Shrinayana. Your slides are not moving.
Only first slide is visible. You are not sharing your.
They are not moving.
I'm moving from my side.
So here it is not visible on the screen.
We are seeing first slide.
Only first slide is visible.
At this point,
You can PPT mode or slide.
You have not, I think, open in presentation mode or slideshow mode.
First slide indicating your title and name is visible to us. Other slides are not visible. Not being shared.
Is it visible now?
No.
Slideshow.
Only first slide is being visible. You just click next on your screen so that the next slide will be visible.
Otherwise, stop sharing and again reshare.
Yeah, you can stop sharing and again reshare it.
28.25.
Again, share it. You should not put off your video. Srinayana.
Sir, I haven't put off my video, sir. It is
26.75.
During the competition, you should not put off your video nor your audio.
okay, sir.
Roll number 1630. Roll number 18.
Yeah, now it is visible. Now the fourth slide is visible. Your video, you start your video. Your video is not visible. Srinena, your video is not visible. Your slides are visible now. Yeah. Your slides are visible.
Sir, is it moving?
Yeah, now it is visible.
Sir, is it moving?
Yeah, you continue. You're wasting your time.
Sir, I'm changing the slide. Sir, I'm changing the slide. Is it moving?
You continue. Slide is visible now. You continue.
27.75. Can I start from the beginning?
No. 25.5. You continue.
These were the trichoderma isolates we have used in the study, and this is a fusarium isolate we have used in the study. And initially, we have screened the trichoderma against fusarium, the dual plate method given by the Dennis and Webster, and it's shown that. Hello, sir.
Srinena, you just click on your screen so that the next slide will be visible to us. You have to do it simultaneously while speaking. You have to click on your.
Sir, I'm changing, sir. I'm changing from my side, sir. But I don't know why it is not visible. I'm able to change from my side.
23.25. Not 3.25. And others are requested to kindly mute their mic, please.
Who is that Aditya Bhatia? Please.
Please mute your mic.
Aditya Bhatia, please mute your mic.
They are disturbing you.
25.50. Chairman, sir, I will do one thing. Otherwise, I will call the next speaker by the time she.
By the time she will give, we'll give another chance to her.
Yeah.
So.
21.25.
Sorry, Srinena. Again, you check your organizing secretary. We will give you a chance. Now I will call on the next contestant, Venkat Ramesh.
Sir, yes, sir. Am I audible, sir?
Ramesh,
yes, sir.
Share your screen.
Yes, sir.
Others are requested to kindly mute their mic. They are disturbing the contestants. Please.
59. Yes, sir. I have shared my screen, sir. Is it visible, sir?
Yes, it's visible.
Sir, am I audible correctly?
Yes, audible, and your slides are visible. Please proceed.
Okay, sir. A pleasant afternoon to each and everyone. I welcome you to my presentation. It's my privilege to present for this PR Verma Award competition for MSc year 2020. So I thank the organizing committee to give me this opportunity in this IMPP first annual conference. So my presentation title goes like characterization, host range, and virulence analysis of bipolar recitaria causing the brown top millet leaf blight in India. So firstly, millets, these are the super grains which we are consuming since the ancient times. Compared to the cereals like rice and wheat, which we are consuming exceptionally in the large quantities, so the millets are very less expansive and nutritionally superior by having those high protein, vitamin, fiber content. So there is no less with the minerals in millets, as the millets are rich in iron, calcium, magnesium, and so they can serve for the nutritional security. And so the FAO, they have considered these millets not only as the smart foods, but also as the smart crops. Smart food means they are having high mineral content and also they are gluten-free, lowest glycemic index, and also high fiber. And as well as these are the smart crops as they are having so many characteristics like photoinsensitivity, climate resilience, drought tolerance, and also they can grow on the marginal soils with less water and less fertilizers. So there are so many initiatives that have been taken up to promote these millets importance for their use and their superiority. So India, we know that India has already announced that 2018 year as a national year of millets, and also FAO approved the India proposal as the 2023 as the international year of millets. And even in India, there are initiatives that are being taken up, as the Odisha has introduced the millets in the public distribution system, like the midday meals and also integrated child development services. So there are some diseases that are destroying the millet crops by reducing their potential yield. So firstly the blast, and also later comes the brown spot and leaf blight. This leaf blight and or brown spot, it is having the historic importance in the other crops like rice, which it causes Bengal famine, and also in the maize, as it causes already southern corn leaf blight, as we all know. And then follows the other disease like rust, alternaria leaf blight, smut, dino mildews, and also other some bacterial diseases. So my objective of the study is like, as this disease brown top millet leaf blight was recently observed in the millet cropping system, like recently in the 2018, it was observed in the germplasm evaluation trains in the ASRP small millets station, Bangalore. So we have took this study as objectives framing like we have to identify and characterize the causal organism of brown top millet leaf blight, and to study their host diversity and also their virulence analysis. So the symptomology of this disease is goes like, the initial symptoms are observed as a small brown spots which is having a little yellow halo, and these initial spots are expressing at 25 to 30 days after sowing. And also as the disease it gets severe, that leaves all the lesions, it is coalescing and getting a blight appearance. And also these disease symptoms is we can able to observe on the leaf sheath at a soil line and also at a boot leaf stage, but there is no symptoms on the glooms or the seeds. And based on this scale, 1 to 90 scale given by the Palana and Das, we have we designed this scale for brown top millet leaf blight, and based on this scale, we have surveyed the 12 different regions major in the major millet growing regions of India that belongs to 5 different states. Among those regions, the Bengaluru of Karnataka has accounted the more like highest disease grade, and followed by the Berhampur of Odisha. Then the Mandya of Karnataka has accounted the least disease severity. So we have collected all the disease samples from different states, and then we have observed under the microscope. Then we carried the isolation and pure culture, and then we have proved the pathogenicity where the symptoms are similar with the original. And also by comparing re-isolated, we have fulfilled our coach postulates, and also we have collected the electronic microscope images to know the hyla structure, which is a characteristic feature of the Helminthosporum species. So based on these morphological characters, we can able to identify the disease, this pathogen that belongs to one of the category that of bipolaris genus. Likewise, we have isolated and recovered the 9 isolates from 5 different states, and carried the virulence analysis where by maintaining one of the control, and observations took a 3 days interval up to the 12 days, where the isolate 3 and 5, it showed the earliest infection at 2 days after the inoculation, but the final disease was covered for the 5th isolate, as all the isolates showed the symptoms which is typical to the brown spot or leaf blight. So this isolate, 5th isolates, was proved to be the more virulent. And then we carried for the morphological characterization on the Zepagdox agar medium. So these are the cultural morphology and also the conidial characters of these 9 isolates. And this is the culture morphology of the 9 isolates, where we have covered days to full cover plate and full pigmentation, colony color, surface and topography, margin, mycelial texture, density, and also the biomass on Zepagdox broth. So as a virulent isolate, this fifth isolate, BTMH5, it accounted for the earliest cover days. It took 6 days to cover the full plate and also the highest biomass. So as it is difficult to discriminate the species of bipolaris by using the morphological characters, we went for the molecular characterization using that CTAP protocol that have been given by the Murray and Thompson in 1980. So we have isolated the DNA and then did the PCR amplification with the two conserved regions of the fungal. So these are the universal, one of the universal primer ITS and also the LSU. So and then we sequenced it, blasted it, and analyzed it phylogenetically, where using the ITS, it is showing all these isolates falling into one category where Bipolaris citaria, Euphorbia, and Emidae is having. So this ITS and also the LSU cannot able to differentiate within the species, but it can able to differentiate only within the genus for these Bipolaris. And then we have carried another analysis by using the heat mapping, using heat mapping version 3.0, where similar results we also found. And then we have carried for another gene region, which is an RPB2, RNA polymerase 2, subunit 2. So as there are no much sequences that are available in the NCBI gene bank regarding to the Bipolaris citaria and Bipolaris emidae, all these 9 isolates, they have formed into a separate category where Sorokinian and Medes all falls into another clade. So this has also visualized as a DNA heat mapping, as you can see. And then we have further moved to for the another gene PCR amplification of the glyceraldehyde 3-phosphate dehydrogenase. For by using this, we can able to differentiate within the species as all the isolate fall into another category, Bipolaris citaria. So, and then we have moved further with a multi-gene approach. So using this, we have collected all the CBS strains information, and we have included ITS, GPDH, and LSU sequences, and then we have concatenated using the MegaX software. And phylogeny is constructed by using the maximum likelihood approach with 1,000 bootstrap replicates. So it shows the results of all the 9 isolates fall into another clade, into one clade where the Bipolaris citaria strain CBS 141.3 was there. So this confirms that the causal organism that is causing the brown top millet leaf blight is the Bipolaris citaria, and this is the first confirmation in India, and first record in India, and also the first confirmation using the molecular characterization from the old. So using these genes, this gene GPDH is proved to be the best marker to discriminate the species level for the Bipolaris genus. And then we have carried for the host strain studies within the small millets to know that what are all the host it is going to infect, but it is shown that only to be infecting brown top millet. So the outcome we have got, we have mapped the disease in several disease major millet growing areas, and can able to identify the organism as Bipolaris citaria characterized, and then isolated the virulent, identified the virulent isolate, and also their host diversity. So the future, in future work, they should be comparative genomics and transcriptome studies. Yes, I want to acknowledge the Equipon Small Millets and also Department Plant Pathology and to the advisory committee. And thank you for your love and support, for your patience. Thank you.
Thank you, Ramesh.
So just please note down, he has taken half minutes extra.
Okay. So
Sir, please
now,
proceed.
Thank you. Now I will call on the next contestant, Nagesh. Nagesh.
Yes, sir. Present, sir.
Yeah. So share your screen.
Yes, sir.
Nagesh, the screen is not visible to us.
Yes, sir. Is it visible, sir?
Not now. Now.
Sir, is it visible now, sir?
Yeah. It's now visible. And Nagesh, your time starts now.
Yes, sir.
On the video.
Okay, sir. Myself, Nagesh. I'm from the Department of Plant Pathology from College of Horticulture, Bagalkot. I have made
Put on your video.
Yes, sir.
And
Yeah, put on your video, Nagesh. Video is not visible.
Okay, sir. Okay, sir.
And slide show ಇದು ಮಾಡಿ. Slide show
Slides are visible, but your video is not started. You start your video.
Nagesh, go to the slide show.
Yes, sir. Yes, sir. Yes, sir. I'm doing now.
Start your front camera.
Video.
Video is not visible.
Sir, is it, sir? No. Now I am going to share the screen, sir. Sir, is it visible now, sir?
Yeah,
It is visible, but your video is not visible.
Video. I have made video.
Nagesh,
On the
slide
videos.
presentation mode ಹೋಗಿ Nagesh.
Okay.
Yeah, now your video is also visible. And your screen is also visible. Nagesh?
While I am sharing screen, the screen is. Vandana? Screen share.
Sir, if Sri Nayana is ready, then you can call Sri Nayana, sir.
Yeah, Nagesh, you get yourself ready. So by the time
Yes, sir, sir, is it, sir?
No, dear, we are having constraints of time.
Sir, is it?
Is it? Yeah, but your video is not
Sir,
visible now.
Am I visible, sir?
No, your
No,
video is
no.
not visible, Nagesh.
You
come
get
and-
yourself
okay, sir,
ready.
sorry for that.
You get yourself ready so that we can finish up the earlier one contestant has failed due to this net connectivity. So, Srinayana,
Yes, sir.
are you ready with your screen sharing and video?
Yes, sir.
We are giving you the second chance for your presentation.
Okay, sir.
Are you ready now?
Yes, sir. I'm ready.
Share your screen. Put on your video and share the screen. Yeah. Your video. The video is not started. You just start your video. Yeah.
Sir, is it visible?
You just change your position because the backlight, which is glowing, it's
Sir, slide is changing.
the light which is focused on the back wall.
You
Yeah,
put on the slide
yeah,
show.
you put, yeah. Now it's okay. So you can.
Sir, slide is changing.
Yeah,
No,
you are
slide
audible.
You put on the slide show mode first.
You
Sir, I
on
have
the
put on the
slide-
slideshow.
No, on your screen there is one presentation like this one is slide share. So you have to put on that one, slide show. One screen like second, third button on the right corner on your screen.
Sir, I have put on the slideshow mode, sir. Is it visible?
You have shared the screen.
Okay,
So
sir.
by looking your screen, we are saying that you are not started the slideshow. Slideshow button is there, just like the screen.
Sir,
In
is
the
it okay now?
bottom right corner, there is one icon for the slideshow. You have to click on that one.
Yes, sir, I have clicked on that one and it is showing slideshow in my computer.
Okay, now it is visible then. Now you can start your presentation.
Okay, so good afternoon to one and all present here. Myself, Srinayana from TNAU Coimbatore. I'm here to present my PR Verma award competition entitled on Trichoderma conjugis mediated interaction of resistant and suppression of fungal limited complex of cucumber under protected cultivation. So as we all know, cucumber is one of the most important vegetable crop which is grown worldwide. India stands fourth position in production and productivity of cucumber apart from Russia, China and Turkey. The cucumber is hampered by many pathogens of which the fungal nematode complex caused by the Fusarium oxysporum cucumberanum and root node nematode Melodogenia incognita plays a major role with 70% crop loss with 10 to 50% reduction in the yield. And here come why we have chosen this topic. When we grow cucumber under protected cultivation for many years, it results in the soil exploitation due to the indiscriminate usage of fungicides and other chemicals, which later on leads to the inoculum development, thereby making pathogens and other components in the soil, leading to the poor soil health due to the lack of beneficial microflora. So when we apply biocontrol agent to this soil, it results in the improved soil health by boosting the immunity of the plant, thereby it results in the increase in production and productivity of the crop. And here comes the objective one of the study. And we have screened trichod. We have used this trichoderma as well as the Fusarium isolates for the study. And we have screened the trichoderma against Fusarium oxysporum cucumberanum. And you can see this.
Your slide, you just share your slides. Slides are first slide is only visible.
Sir, I'm changing from my side, sir. I don't know this.
Only first slide is visible to us.
Some other system.
Sir, I'm in 7th slide, sir.
No, it is the first slide only. It is visible here.
So can I do in this editing mode, sir?
Your slides are not moving, and unless your slides are visible to us, we cannot. The judges cannot judge you.
PDF mode.
Okay. Sir, can I move in editing mode, sir?
No, you just convert the slide into PDF and present.
Oh, okay, sir.
PowerPoint in the save model.
Yes, convert is PDF.
Okay, try now.
So, Nagesh, are you ready by this time?
Nagesh?
Nagesh?
Sir, Chairman, sir.
Yes, sir. Yes, sir.
Are you ready to present your presentation for this competition?
Yes, sir. Yes, sir. Yes, sir. Definitely, sir.
Yeah. So share your screen.
Okay, sir. I am automatically off again.
Srinana, by the time you can convert your presentation, the PDF, and get ready, because after 10 minutes, you have to, after 10 minutes, you have to present your presentation. Because this is the, Nagesh is the last contestant, and after that, you can give only the, after 10 minutes, you have to be ready for the presentation. Okay? Nagesh, can you share your screen?
Sir, what happens? Sir, problem is with the while I'm sharing the screen, the video will putting off, sir. Automatically.
You are joined from your mobile or laptop?
Laptop, sir.
Laptop, no, then your front camera will start, no? By the front camera, your video will be open. You can share your screen.
Yes, sir. Okay, sir.
You open in PPT form. Because there are one green option is there, share screen. You have to click on that share screen.
Yeah, yes, sir. Yes, sir.
You just click on your share screen.
Sir, Chairman, sir, it seems both of them are having some serious problems. We will do like this. We'll break for lunch now, and we will assemble at one. I mean, exactly at 2.
No,
It is
sir.
already 1:20.
Yeah, by the time these.
Now it's,
Nagesh,
sir, please,
Nagesh,
sir.
yeah, Nagesh is possible.
Yes, sir. Is it visible, sir, now?
Share is this one, but your video is not visible here. Nagesh,
Still,
your
I have
laptop
made a
camera,
video.
your laptop camera is being this one. Yeah, now it is. That video camera is also visible. Screen is also visible.
Yeah, thank you, sir. Thank you, sir.
Organizing
Yeah,
secretary,
okay, sir. Okay, sir.
I will take the permission from the organizing secretary.
Okay, okay.
Sir, let me proceed. It is now visible.
Yeah, yeah. So Nagesh,
okay,
you can proceed.
Sir, sorry for disturbance. Sorry for disturbance, sir. Myself, Nagesh, from Department of Plant Pathology, College of Horticulture, Bagalkote. I have did a research work on studies on variability and alternate solani, which is causing by early blight, which is caused early blight disease in a tomato. So we all of you know the tomato is an important cash crop, which is botanically called as Solanium lycopersicum, which is the chromosome number is 2 and 24. The comes under family is Solanaceae, having the origin of Andean region of South America. We all of us know the huge post-harvestical concept. If you consider post-harvest concept, the huge importance of post-harvestal losses in a tomato. So we can make a puree, paste, soap, juices, ketchup, and we can also use for canning in canning industry for these attractive fruits. This also has a high medicinal values with a rich source of vitamin A, vitamin B, C, and rich source of minerals. And it's also called in poor man's apple. Coming to area production and productivity, our India stands second in area production with 458.24 million hectares with an area production with 15.22 million tons production with a productivity average national productivity is 32.8 tons per hectare. With an area of 578,000 hectares with an area. Yes, sir.
Your
Yes,
slides,
sir.
you have same problem like Srinaya. Your slides are also not moving.
Sir.
Only first slide is.
Sir, is it, sir?
Now moving.
Sir, now.
Presentation, bring
Now
down
this second. Yeah. You are sharing second slide too. Okay. Continue.
Yes, sir. Yes, sir.
Continue.
Yes, okay, sir. As tomato is a host highly susceptible to different types of fungi, bacteria, viruses, nematodes, and phytoplasma, different types of diseases which can be too prone to any biotic and abiotic stresses. Among the biotic stresses, the fungal diseases are the most prominent. So early blight, which is caused by an alternate solani, late blight, Phytophthora infestans, damping of pythium, bacterial wilt, fungal wilt, and the most devastating diseases like tomato leaf curl virus and nematodes like Melgogena incognita. Coming to taxonomic position of an alternate solani, which comes under kingdom fungi, phylum Ditromycetes, class Dothromycetes,
Dear,
subclass
at the same time, you share your screen. Next slide. Yeah, because your
I have to go.
Presentation and the slides, they are not matching. Kindly give attention
yes, sir. Because
one.
of this. Yes, sir. If I made a presentation mode, that disappears. In my laptops, it will be visible, sir. Comes under family Pleosporiaceae, under genus alternate species Solani. Coming to epidemiological life cycle of an alternate Solani, the mycelia and condia which overwinters in a natural ecosystem, which can make a color rot in seedlings. The matured lesions which produce the conidiophores and conidia produced from matured lesions, which are dispersed by wind and rain splash. The matured conidia which go and infect a healthy tissue that makes a similar symptoms as the target eye or bullseye symptoms on a fruit or petioles or stem or leaves. These are the different symptoms which has been observed on the leaves, we can find the target spot symptoms on a leaf within concentric rings within yellow halo. On a leaves, the stem we can cankerous growth on a stem within raised these things with a black color spore masses we can see on fruits. And this is in culture of an alternate solani. This is a single muricate conidia with a transverse and vertical septation. Coming to major objectives, the first objective is survey for assessing the disease severity of an early blight. The study is to study the variability among the collected isolates of an alternate solani, to evaluation of tomato different genotypes against early blight disease, the evaluation of fungicides against early blight disease. Coming to as we know, the pathologist, we have to prove the pathogenicity. So isolation, the identification, and proving the pathogens of an alternate solani. These are the 1 month old plant seedlings have inoculated with a spore suspension, incubated for 2 to 3 days. We can observe the symptoms within concentric rings. We have made the survey within major southern part of Karnataka. The major growing parts like major diverse hot belt of this tomato, what we are called as Tumkuru, Chikbalapura, Bengaluru rural, Kolar, Ramnagara, and Chamarajnagara. So with the help of Datar and May, they have given some scale 0 to 5 scale. In 1986, they have made with a 0 to 5 scale with the help of Wheeler has made the formulated formula with a percent disease index. With the help of that, we can score the host resistance, host reactions. Based on host reactions, we can note that percent disease indexing. So among them, the Chowdenhalli in a village in Kolar Taluk have been observed by 57.50, and with the least which in Kumankunte of Srinivaspura of the same Kolar district. In Chikbalapur, the Ulonayakanahalli in Chintamani Taluk having the least disease severity when compared with the similar district, the Bachhadi, which shows the maximum disease severity. In case of Tumkur, Rayavara village, which shows the maximum disease severity with 35.75 compared followed by Hegeri. The Hegeri, which shows the least disease severity. In Ramnagara, Hallimala in Ramnagara Taluk, it shows the high disease severity, which is followed by the least severity is in Harisandra of Magadi Taluk in Ramnagara district. Coming to the district wise, if you compare with the district wise mean severity of the alternate solani, so Kolar, Chikbalapur, Tumkur, Ramnagara, we have noted now the maximum disease index in case of Kolar district with the 50.43. Followed by Chikbalapura 42.75, Tumkur 31.75, and Ramnagara. This is why because of continuous monocropping in case of Kolar Chikbalapur disease, which makes a severe epidemic disease. So it hence we are with the different host susceptibility and different environmental characters and epidemiological factors, we can note at the maximum disease severity in case of Kolar. So coming to variability studies, we have made we have collected the 9 different isolates from throughout the Karnataka, from Bidar district, Bagalkot, Kolar, Chikbalapura, Tumkuru, Ramnagara, Chikmagaluru, Bengaluru, and Mysuru. We have collected these 9 different isolates, and we have tried out the different solid agar media like potato dextrose agar, and we have observed different colony characters like colony color, mycelial growth, margins of the colony, and zonations. And we have also tried on Richard's agar, which shows the whitish with gray centers, the gray with the dark gray centers. So the mycelial growth may be cottony or slightly cottony. The margins of the colony is round with the whitey margins. The zonations may or may not on that time. The slightly zonations or there is no zonations, or in some isolate which shows the zonations. These are the potato dextrose agar plates. These are the Richard's agar. These are the alternate solani which has been characterized on subordinate dextrose agar. Often the Bidar isolate which shows a gray color within compact or some isolate which showing the cottony growth or sparse growth. The margins of the colony is irregular to regular. The zonations, again we have noted zonations. These are the subordinate dextrose agar and V8 juice agar. Coming to Zabax dogs agar, we have tried the colony color is gray color with a raised pinkish white at its center. Some having the mycelial cottony or slightly cottony, irregular zonations with or without zonations. These are on the different radial growth or cultural characteristics on in Zabax dogs agar. These are the variability in a radial growth of different isolates of an alternate solani. If you compare with the different isolates, we have, irrespective of any isolates, we have got a maximum radial growth in case of Zabax dogs agar, followed by Richard's agar and Zabard's dextrose agar. And the least which has been growth which has been found in protected extracellular. The variability in growth of different isolates. So the Bagalkote isolate which goes the average mean, if you consider the Bagalkote isolate which gives the maximum mean length with compared with the Chikmagalur which is the followed by in Chikmagalur, and the least growth which has been noted in Kolar district. Coming to different spore morphological studies, these are the different spore morphology from in different isolates. When comparing with the spore morphology, having the number of transfer separations, a number of longitudinal separations, the length of the conidia, the beak length, the width of the conidia. So number of transfer separation is maximum in case of Bangalore, which is the least which is found in Ramnagara, and the number of longitudinal separations which has been more in case of Ramnagara, which has been least in case of Bidar, and the length of the conidia within beak, the maximum length of the conidia within beak, Chikbalapur isolates followed by in Tumkur, the beak length which has been 5.2 within in Ramnagara isolates, followed by the least which has been found in Kolar district, the width of the conidia which has been maximum in case of Kolar district, the minimum or least width of the conidia in Bidar isolate. These are the transplanted seedlings. After 50, we have made the screening experiment for 50 genotypes, which we have compared with Czech isolates, Arca Vikas, transplanted 50 different genotypes which are collected from Department of Vegetable Science, College of Horticulture, Bangalore. These are the 15 days and 1 month old seedlings plants. These are overlook of screening of tomato genotypes. These are the 50 genotypes screened. After 30 days, 60 days, 90 days after screening, we have made the attempt in host reaction response. We have noted down within different yield parameters. Yield parameters we also took. So after that screening, we have made the host resistance, and we have categorized the different genotypes based on host response. So we got 10 different genotypes with a resistance, and 30 different genotypes with a moderate resistance, 9 within susceptible, 1 within highly susceptible, which is COHB 143. These are the different host response we can see morphologically. These are potato leaf types which shows the disease resistance against alternate solanine. These are the moderate resistance 47, 48, 40, 68 genotypes. These are the susceptible plants within 46, 48, 70 genotypes. These are highly susceptible. We can compare with highly susceptible, and this Arca Vikas is used rather than susceptible check. Coming to in vitro, we have made the last management purpose. So we have made the in vitro efficacy of fungicides against alternate solanine. So we have made the 11 different triazoles, benzimidazoles, and strobilurins compounds. Among those, we got a 70% result at a least minimum concentration at a 25% of 25 ppm. We got a maximum percent inhibition of a mean inhibition in case of tebuconazole, difenoconazole, which is followed by strobilurins compounds like tebuconazole, trifaclostrobin, which is called a nativo, metrium plus pyraclostrobin, which is carburetok. These are the effective fungicides we have evaluated, and followed by Zineb. Zineb, which gives Zineb, and Mancozeb, which gives the Zineb, Mancozeb, Dodaine, and
your time is up. You have to wind up.
Least growth. And these are the overview of a field experiment. These are the what we have effective fungicides we have taken out to the field experiment. The difenoconazole, tebuconazole, which shows the higher efficacy, which means the percent disease controls within 64% within difenoconazole, followed by tebuconazole, and with an yield of 30.07 tons of ton per hectare. The tebuconazole, we can see that the morphological difference between tebuconazole sprayed and control plants. There is a wide range of this is a summary of the rowing survey, which gives the Kolar, Chikbalapur, Ramnagara districts, which shows the higher disease, which followed by the 50.4, which is more disease severity in case of Kolar, followed by Tumkur. The Zapax doxagar, which shows the common, which is the average growth of which gives the highest growth of fungus. The 50 genotypes around, we have categorized the different based on host response. The difenoconazole, tebuconazole, which shows the higher in vitro efficacy. And in case of field, the tebuconazole, trifaclostrobin, native, and pyraclostrobin, and cabrate, this is metrium, which gives the 30.07 ton per hectare within a maximum good yield. Thank you, sir.
Sir, he has taken 1.5 minutes extra. Judges, please note down.
So now the last contestant, Srinena.
Yes, sir.
Are you ready with your presentation?
Yes, sir. I have made into PDF.
You made it. So, are you able to start it now?
Yes. Sir, is it visible?
You share your screen so that
Sir, is it visible?
It is not visible.
your screen is visible. You just share your presentations. You just click on your screen. Whatever your PDF is visible, you just double click on that one.
First open the file.
Yeah, open this file. You just double
And
click.
after that, go to the sharing option. Outside, you first open the slide or the PDF, then go to share option.
With the help of your mouse cursor, you just go on this screen, which is Srinarayan BR Parma Award Adobe PDF. So you double click on that slide so that it can be opened.
It is open, sir, but don't.
Organizing secretary, sir, can we allow her after lunch time?
Yes, sir. Yes, sir.
So by the time she will take technical assistance from somebody and know how to start this presentation and how to present it.
Srinayana, make sure that it is ready after lunch.
Okay.
You will be given
Okay,
another
sir.
chance.
Okay, sir.
It is not problem at our side. Please be in the where the net connectivity and all is good, and you just make some rehearsal and be ready by 2 o'clock.
Okay, sir.
Sir, Chairman, sir.
Sir Kubbali, whether we have to join by 2 or?
Yes, madam, you have to rejoin by 2.
Okay, okay.
Sir, Chairman, sir, is it okay, sir?
Sir, it's only 15 minutes, 20 minutes hardly we are getting for taking our food. So kindly extend by 15 minutes at least.
Sir, by 2:15 shall we start?
Yeah, 2:15 so that at least we can have some
Okay,
food.
sir. Okay, sir. Participants, please rejoin at 2:15.
Okay, thank you, sir.
Thank you, sir. Thank you.
Thank you, Kubbali.
Thank you, madam.